Hello there, and welcome to Midnight Sleep Science. Right now, in this very moment, something extraordinary is happening inside your body. Without thought or effort, billions of specialized cells are translating the physical world into electrical signals your brain can understand. Light is striking the back of your eyes and becoming vision. Air molecules are vibrating tiny bones in your ears and becoming sound. Pressure against your skin is activating receptors and becoming touch. This silent symphony happens continuously, translating reality into experience. Most people believe they have 5 senses, but the truth is more remarkable. Beyond sight, sound, taste, smell, and touch, your body possesses hidden senses that tell you where you are in space, whether you are moving, and even the position of your limbs without looking. Together, these sensory systems create something science still struggles to fully explain: your conscious awareness of being alive. Tonight, we will explore the real mechanisms behind how your body understands the world, not through imagination or metaphor, but through the actual cells, pathways, and structures that make perception possible. The journey will be gentle, factual, and grounded in what truly exists, because the reality of how you sense your world is already beautiful enough. If you enjoy these gentle journeys, feel free to like and subscribe. But for now, let's begin. Every moment of your life, your body is listening to the world, not with your ears alone, but with billions of specialized cells scattered throughout your tissues, each one designed to detect a specific type of change in your environment. These cells are called sensory receptors, and they are the reason you know anything at all about the world around you. Without them, there would be no sight, no sound, no sensation of warmth or cold, no awareness of touch or pain. You would exist in complete sensory silence, disconnected from reality. Right now, sensory receptors in your skin are responding to the pressure of whatever surface you are resting on. Receptors in your eyes are detecting the light in the room, even if your eyelids are closed. Receptors in your inner ear are monitoring the position of your head in space. All of this happens without conscious thought, without effort, without your awareness. These cells are constantly active, constantly translating the physical world into a language your nervous system understands. The process begins with something scientists call a stimulus. A stimulus is simply any form of energy that can be detected by a sensory receptor. Light is a stimulus. Sound waves are stimuli. Pressure against your skin is a stimulus. The molecules that create smell and taste are stimuli. Even the pull of gravity on the fluid in your inner ear is a stimulus. Each type of receptor is designed to respond to one specific kind of energy, and when that energy reaches a certain threshold, the receptor activates. What happens next is one of the most elegant processes in all of biology. The receptor converts the physical stimulus into an electrical signal. This conversion is called transduction, and it is the foundation of all sensory experience. Light becomes electricity, sound becomes electricity, pressure, temperature, chemicals, all of it becomes electricity. Your entire perception of reality depends on this transformation. This translation from the language of physics and chemistry into the language of neurons. The electrical signals generated by sensory receptors are not like the electricity that powers your home. They are created by the movement of charged particles called ions across the cell membrane. Every cell in your body maintains a difference in electrical charge between its inside and outside, a state called the resting membrane potential. When a sensory receptor is stimulated, special channels in its membrane open, allowing ions to flow in or out. This flow of ions changes the electrical charge inside the cell, creating what is called a receptor potential. If the receptor potential is strong enough, it triggers an action potential, a rapid electrical impulse that travels along a sensory neuron toward your spinal cord and brain. This is how information moves through your nervous system, as waves of electrical activity passing from cell to cell, neuron to neuron, until the signal reaches the regions of your brain that can interpret it. The speed of this transmission varies depending on the type of neuron. but some signals travel at over 200 miles per hour. Sensory receptors come in many forms, each specialized for a different task. Photoreceptors in your eyes contain molecules that change shape when struck by light, initiating the process of vision. Mechanoreceptors in your skin have structures that deform under pressure, opening ion channels and creating electrical signals. Chemoreceptors in your nose and tongue bind to specific molecules, triggering responses that your brain interprets as smell and taste. Thermoreceptors detect changes in temperature. Nociceptors respond to potentially harmful stimuli, creating the sensation of pain. What is remarkable is that despite their diversity, all sensory receptors operate on the same basic principle. They detect a stimulus, they transduce that stimulus into an electrical signal, and they transmit that signal to the central nervous system. The difference lies in what they are tuned to detect and where in the brain their signals are sent. A photoreceptor sends its signal to the visual cortex. A mechanoreceptor in your fingertip sends its signal to the somatosensory cortex. Your brain knows what you are sensing, not because the electrical signals themselves are different, but because of which neurons are firing and which pathways they travel along. This concept is labeled line coding. Every sensory receptor is part of a specific pathway, a dedicated line of communication between a particular location in your body and a particular region in your brain. When neurons in the visual pathway fire, you experience sight. When neurons in the auditory pathway fire, you experience sound. The information is identical in form, electrical impulses, but the meaning is determined by the route it takes. Sensory receptors also vary in how they respond over time. Some receptors adapt quickly, meaning they fire intensely when a stimulus first appears, but then reduce their activity even if the stimulus continues. This is why you stop noticing the feeling of your clothes against your skin after a few moments. The receptors detected the pressure when you first put the clothes on, but they adapted, reducing their signaling because the stimulus is constant and not new. This allows your nervous system to focus on changes in your environment rather than being overwhelmed by unchanging background information. Other receptors adapt slowly or not at all, continuing to send signals as long as the stimulus is present. Pain receptors often fall into this category. If you place your hand on a hot surface, the nociceptors will continue firing, sending urgent signals to your brain until you remove your hand. This makes sense from a survival perspective. Pain is a warning, and the warning needs to persist until the threat is gone. The sensitivity of sensory receptors is also variable. Some receptors have very low thresholds, meaning they can detect extremely subtle stimuli. The photoreceptors in your eyes, for example, can respond to a single photon of light under ideal conditions. Other receptors require stronger stimuli to activate. This range of sensitivity allows your sensory systems to function across an enormous range of conditions, from dim starlight to bright sunshine, from the faintest whisper to the roar of thunder. Your body contains millions of sensory receptors, distributed throughout your tissues in patterns that reflect their function. Your fingertips, for instance, have a much higher density of touch receptors than the skin on your back. This is why you can detect finer details with your fingers, why you can read Braille or feel the texture of fabric so precisely. The receptor density determines your sensory acuity, your ability to distinguish between two closely spaced stimuli. In your eyes, photoreceptors are concentrated in the retina, particularly in a small central region called the fovea, where your visual acuity is sharpest. In your ears, thousands of hair cells line the cochlea, each tuned to a different frequency of sound. In your skin, receptors are embedded at different depths, each type positioned to detect specific types of mechanical or thermal information. This distribution is not random, but reflects millions of years of evolutionary refinement. The architecture of sensory receptors is breathtaking in its precision. Each receptor is a tiny machine built from proteins and lipids, assembled according to instructions encoded in your DNA. The ion channels that open in response to stimuli are themselves complex structures, with gates and filters that control which ions can pass through. The molecules in photoreceptors that respond to light are so efficient that they can change shape in less than a trillionth of a second. Every aspect of these cells has been shaped by evolution to perform one task with extraordinary efficiency: to sense. And yet, despite their complexity, sensory receptors are vulnerable. They can be damaged by disease, injury, or age. When photoreceptors degenerate, vision fades. When hair cells in the cochlea are destroyed by loud noise, hearing diminishes. The loss of sensory receptors is often irreversible, because many of these cells do not regenerate in adult humans. This fragility makes them all the more precious, all the more worthy of protection. What makes sensory receptors truly remarkable is not just what they do, but what they make possible. They are the interface between your inner world and the outer world. They are the first step in the long chain of processing that leads to conscious experience. Without them, your brain would be silent, isolated, cut off from reality. With them, your brain has access to information about light and sound, touch and taste, movement and position. They are the gateway through which the universe enters your awareness. The journey from receptor to perception is not instantaneous. When a sensory receptor generates an electrical signal, that signal must travel along a sensory neuron, sometimes across considerable distances. A touch receptor in your toe, for example, sends its signal all the way up your leg, through your spinal cord, and into your brain. This journey can take a fraction of a second, but in neural terms, that is a significant amount of time. The sensory neurons that carry these signals are specialized for transmission. Many are wrapped in a fatty substance called myelin, which acts as insulation and dramatically increases the speed at which electrical impulses can travel. The myelin is not continuous, but forms segments separated by small gaps called nodes of Ranvier. The electrical signal jumps from node to node in a process called saltatory conduction, allowing it to move much faster than it would along an unmyelinated fiber. This design is one of the nervous system's most elegant solutions to the problem of rapid communication over distance. Not all sensory neurons are myelinated, however. Some, particularly those that carry information about dull, aching pain or temperature, are unmyelinated and conduct signals more slowly. This is why you sometimes experience a delay between an injury and the full sensation of pain. The sharp, immediate pain you feel when you stub your toe travels along fast myelinated fibers called A-delta fibers. The slower, throbbing ache that follows arrives via unmyelinated C fibers. Your nervous system uses different speeds of transmission for different types of information, prioritizing urgent signals while allowing less critical information to arrive more slowly. As sensory signals travel toward the brain, they pass through relay stations where neurons connect and communicate. The first major relay point for most sensory information is the thalamus, a structure deep in the center of your brain. The thalamus acts like a sorting center, receiving incoming sensory signals and directing them to the appropriate regions of the cerebral cortex for processing. Almost all sensory pathways pass through the thalamus before reaching the cortex. The one exception is smell, which has a direct pathway to cortical areas, bypassing the thalamus entirely. This unique routing may explain why smells can trigger such immediate and powerful memories. The organization of sensory pathways is precise and orderly. Signals from the left side of your body generally travel to the right side of your brain, and vice versa. This crossing over, called decussation, occurs at specific points along each sensory pathway. For touch and proprioception, the crossover happens in the medulla, a region at the base of the brainstem. For vision, it occurs at the optic chiasm, where the optic nerves from each eye partially cross. This contralateral organization means that damage to one side of the brain often affects sensation on the opposite side of the body. Within each sensory pathway, information is organized topographically. This means that the spatial arrangement of receptors in your body is preserved as signals travel through the nervous system. Adjacent receptors send signals to adjacent neurons, which connect to adjacent regions in the brain. This creates maps of your body surface in the somatosensory cortex, maps of the visual field in the visual cortex, maps of sound frequency in the auditory cortex. Your brain is filled with these maps, representations of the external world, and your body's relationship to it. The strength of a sensory signal is encoded in the frequency of neural firing. A weak stimulus might cause a sensory neuron to fire a few times per second. A strong stimulus causes the same neuron to fire many times per second, sometimes hundreds of times. Your brain interprets these firing rates as intensity. A dim light produces low-frequency firing in photoreceptors. A bright light produces high-frequency firing. The same principle applies to all sensory modalities. Louder sounds, stronger pressures, more intense tastes, all produce higher rates of neural firing. But frequency is not the only code your nervous system uses. The number of receptors activated also matters. When you apply light pressure to your skin, only a few mechanoreceptors respond. When you apply firm pressure, many more receptors activate. Your brain reads both the frequency of firing in individual neurons and the population of neurons that are active, combining these two sources of information to determine what you are sensing. The process of sensation is remarkably efficient. At any given moment, millions of sensory receptors are active, generating electrical signals, sending information toward your brain. Yet you are not overwhelmed by this flood of data. Your nervous system filters, prioritizes, and selects which information reaches your conscious awareness. Much of what your sensory receptors detect never becomes a conscious sensation at all. It is processed automatically, used to regulate your body's functions or guide your movements without your awareness. This filtering begins at the level of the sensory receptors themselves. Receptors that adapt quickly stop sending signals about constant, unchanging stimuli. It continues in the spinal cord, where circuits of neurons can modulate incoming sensory signals, amplifying some and suppressing others. It continues in the thalamus and cortex, where attention and expectation shape which sensory information is processed most deeply. Your perception is not a passive recording of reality, but an active construction, built from selected fragments of the vast sensory information available to your nervous system. The gateway cells, these sensory receptors scattered throughout your body, are doing their work right now. They are detecting, transducing, transmitting. They are the beginning of everything you know about the world. And from this beginning, the real journey of perception begins. As signals travel deeper into your nervous system, toward the regions where sensation becomes experience. The diversity of sensory receptors reflects the complexity of the world they must detect. Consider the receptors in your skin alone. There are at least 6 distinct types of mechanoreceptors, each responding to different aspects of touch. Merkel cells detect sustained pressure and fine details, allowing you to read braille or feel the edges of objects. Meissner corpuscles respond to light touch and changes in texture, firing rapidly when something brushes across your skin. Pacinian corpuscles sense deep pressure and high-frequency vibrations, letting you feel the rumble of a passing truck or the buzz of a phone in your pocket. Ruffini corpuscles monitor skin stretch, providing information about the position and movement of your fingers and limbs. Each type of mechanoreceptor has a unique structure that determines its function. Pacinian corpuscles, for example, are shaped like tiny onions, with concentric layers of tissue surrounding a central nerve ending. When pressure deforms these layers, they compress the nerve ending, opening ion channels and generating an electrical signal. The layered structure acts like a filter, making the receptor especially sensitive to rapid vibrations while filtering out steady pressure. This is why you can feel your phone vibrating in your pocket, but quickly stop noticing the constant pressure of your wallet. The receptors in your eyes operate on a completely different principle. Photoreceptors contain molecules called photopigments, which are proteins bound to a light-sensitive component derived from vitamin A. When light strikes a photopigment molecule, it causes a change in the molecule's shape, triggering a cascade of chemical reactions inside the receptor cell. This cascade ultimately closes ion channels, reducing the flow of ions and changing the cell's electrical state. Unlike most sensory receptors, which become more active when stimulated, photoreceptors actually become less active in the presence of light. This inverted response is one of the peculiarities of vision, a design that has proven remarkably effective for detecting light. In your inner ear, mechanoreceptors take yet another form. The hair cells that detect sound and movement have bundles of tiny projections called stereocilia extending from their surface. These stereocilia are arranged in order of height, like the pipes of an organ. When fluid motion or mechanical displacement bends the stereocilia toward the tallest member of the bundle, it pulls on tiny protein filaments that connect one stereocilium to the next. These filaments act like springs, opening ion channels at their attachment points and allowing charged particles to flow into the cell. The hair cell responds within microseconds, making it one of the fastest sensory transduction mechanisms in your body. The chemoreceptors in your nose and tongue use yet another strategy. These receptors have proteins embedded in their cell membranes that act as binding sites for specific molecules. When a molecule with the right shape and chemical properties binds to one of these receptor proteins, it triggers a signaling cascade inside the cell. This cascade opens ion channels, creating an electrical signal that travels to your brain. Your nose contains hundreds of different types of olfactory receptors, each tuned to detect different molecular features. Your tongue has a smaller number of taste receptor types, each specialized for one of the 5 basic tastes: sweet, salty, sour, bitter, and umami. What unites all these diverse receptors is their fundamental role as transducers. They stand at the boundary between the physical world and your neural representation of it. They convert forms of energy that exist independently of you, photons, pressure waves, molecules, mechanical forces, into the electrical language your nervous system uses. Without this conversion, these physical phenomena would remain forever beyond your awareness. Light would strike your eyes, but you would not see. Sound waves would enter your ears, but you would not hear. The world would exist, but you would have no way to know it. The precision required for this transduction is extraordinary. Your sensory receptors can detect changes in stimulus intensity across a range spanning many orders of magnitude. The faintest sound you can hear represents a movement of your eardrum that is smaller than the diameter of a single atom. The brightest light you can tolerate is more than 1 billion times more intense than the dimmest light you can detect. Your sensory systems operate across these enormous ranges while maintaining the ability to detect subtle differences in intensity. This is accomplished through a combination of receptor adaptation, neural processing, and the logarithmic encoding of stimulus intensity. The reliability of sensory transduction is also remarkable. Millions of times each day, sensory receptors detect stimuli, generate electrical signals, and transmit information to your brain. The vast majority of these transduction events occur without error. The molecular machinery operates with a consistency that rivals or exceeds any human-made device. And when transduction does fail, when receptors are damaged or diseased, the consequences for perception can be profound. A relatively small loss of photoreceptors can significantly impair vision. The death of hair cells in the cochlea leads to permanent hearing loss. Your sensory experience depends entirely on the faithful operation of these microscopic cellular machines. As you rest here, sensory receptors throughout your body continue their ceaseless work. They are detecting the pressure of the surface beneath you, the temperature of the air around you, the dim light that penetrates your closed eyelids. They are sending streams of electrical signals toward your brain, carrying information about your body and your environment. These signals will travel along defined pathways, cross synapses, activate networks of neurons, and from these electrical impulses somehow emerges your experience of being here, now, aware. The gateway cells have opened, the information is flowing, and waiting at the first stop on this journey is perhaps the most studied sensory system of all. The intricate machinery that transforms light into sight. Of all the sensory systems in your body, none has captured human curiosity quite like vision. The ability to perceive light, to see colors and shapes, to recognize faces and navigate through space. This is the sense most people fear losing above all others. And yet, vision begins with something remarkably simple. Particles of light entering a small opening in the front of your eye. Light is electromagnetic radiation, waves of energy that travel through space at tremendous speed. When light reflects off objects in your environment, it carries information about those objects, their color, their shape, their distance from you. Your eyes are biological instruments designed to capture this light and extract that information. The process begins at the very front of your eye with a clear curved structure called the cornea. The cornea is your eye's first lens. It is transparent, allowing light to pass through while simultaneously bending that light inward through a process called refraction. The cornea performs most of the focusing work in your visual system, bending incoming light rays so they converge toward a point deep inside your eye. The curvature of the cornea is precise, and even small irregularities in its shape can blur your vision. A condition known as astigmatism. Behind the cornea lies a space filled with a clear fluid called aqueous humor. This fluid nourishes the tissues at the front of your eye and maintains the eye's internal pressure. Light passes through this fluid and encounters the next structure, the iris. The iris is the colored part of your eye, the ring of muscle and pigment that gives your eyes their distinctive hue: brown, blue, green, hazel. But the iris is not merely decorative. It functions as an adjustable aperture, controlling how much light enters the deeper parts of your eye. At the center of the iris is an opening called the pupil. The pupil appears black because you are looking through it into the dark interior of your eye. The iris contains two sets of muscles that control the pupil's size. In bright light, circular muscles contract, making the pupil smaller and reducing the amount of light that enters. In dim light, radial muscles contract, enlarging the pupil and allowing more light to reach the photoreceptors at the back of your eye. This adjustment happens automatically, continuously, without conscious control. Light that passes through the pupil next encounters the lens. Unlike the fixed shape of the cornea, the lens is flexible, and its shape can be adjusted by small muscles called ciliary muscles. When you look at something far away, these muscles relax, allowing the lens to flatten. When you look at something close, the muscles contract, making the lens more curved. This process, called accommodation, allows your eye to fine-tune its focus depending on the distance of the object you are viewing. As people age, the lens gradually loses its flexibility, making it harder to focus on nearby objects, a condition called presbyopia. After passing through the lens, light travels through the large interior chamber of the eye, which is filled with a clear, gel-like substance called vitreous humor. This substance helps maintain the eye's shape and allows light to pass through with minimal distortion. Finally, the light reaches its destination, the retina. The retina is a thin layer of tissue lining the back of your eye, and it is here that light becomes neural information. The retina is one of the most complex tissues in your body, containing multiple layers of cells organized with extraordinary precision. The outermost layer, the one furthest from incoming light, contains the photoreceptors, the cells that actually detect light. There are two types of photoreceptors, rods and cones. Rods are extremely sensitive to light and allow you to see in dim conditions. You have approximately 120 million rods in each eye, distributed primarily in the peripheral regions of your retina. Rods do not detect color. They provide black and white vision. This is why colors fade when lighting becomes very dim. Only your rods are active, and they cannot distinguish wavelengths. Rods contain a photopigment called rhodopsin, which changes shape when struck by light, initiating the process of phototransduction. Cones are less sensitive than rods, but allow you to see fine detail and perceive color. You have about 6 million cones in each eye, concentrated primarily in a small central region of the retina called the fovea. The fovea is the area of sharpest vision, the point where images are most clearly focused. When you look directly at something, you are positioning the image on your fovea, where cone density is highest. There are 3 types of cones, each containing a different photopigment that responds preferentially to different wavelengths of light. One type responds most strongly to short wavelengths, which you perceive as blue. Another responds to medium wavelengths, perceived as green. The third responds to long wavelengths, perceived as red. Your perception of color arises from comparing the responses of these 3 cone types. When light strikes a photoreceptor, it triggers a cascade of chemical reactions. The photopigment molecule absorbs the light energy and changes shape, activating a protein called transducin. Transducin activates an enzyme called phosphodiesterase, which breaks down a molecule called cyclic GMP. The reduction in cyclic GMP causes ion channels in the photoreceptor's membrane to close, reducing the flow of sodium and calcium ions into the cell. This change in ion flow alters the cell's membrane potential, creating an electrical signal. Remarkably, photoreceptors respond to light by hyperpolarizing, becoming more negative, rather than depolarizing like most sensory receptors. In darkness, photoreceptors are partially depolarized, continuously releasing neurotransmitter. When light activates them, they hyperpolarize and reduce their neurotransmitter release. The cells that receive signals from photoreceptors interpret this reduction in neurotransmitter as the presence of light. The photoreceptors do not send signals directly to the brain. Instead, they connect to other neurons within the retina in a complex network of processing. Photoreceptors synapse onto bipolar cells, which in turn connect to ganglion cells. The axons of the ganglion cells bundle together to form the optic nerve, which exits the eye and carries visual information to the brain. At the point where the optic nerve exits, there are no photoreceptors, creating a blind spot in your visual field. You are typically unaware of this blind spot because your brain fills in the missing information based on surrounding context. The retina also contains horizontal cells and amacrine cells, which form lateral connections between photoreceptors, bipolar cells, and ganglion cells. These connections allow the retina to perform sophisticated processing, enhancing contrast, detecting edges, and beginning the work of feature extraction before visual information ever reaches the brain. In a very real sense, the retina is an extension of the brain, a piece of neural tissue that has migrated forward into the eye during development. The signals that leave your retina through the optic nerve carry a transformed version of the visual world. These are no longer simply representations of light and dark, but processed information about contrast, edges, movement, and color. The ganglion cells that generate these signals have receptive fields, meaning each responds to light falling on a specific region of the retina. Some ganglion cells fire when light hits the center of their receptive field and dim when light hits the surrounding area. Others do the opposite, firing when the surround is illuminated and the center is dark. This center-surround organization enhances your ability to detect boundaries and edges, the fundamental building blocks of visual form. The optic nerves from both eyes travel back into your skull, converging at a structure called the optic chiasm. At the chiasm, something important happens. The nerve fibers partially cross over, fibers carrying information from the nasal half of each retina, the half closer to your nose, cross to the opposite side of the brain. Fibers from the temporal half, the half closer to your temples, remain on the same side. This arrangement means that everything in your left visual field, seen by both eyes, is processed by the right side of your brain, and everything in your right visual field is processed by the left side. After the optic chiasm, the nerve fibers continue as the optic tracts, traveling to a structure deep in your brain called the lateral geniculate nucleus, or LGN. The LGN is part of the thalamus, the brain's sensory relay station. Nearly all visual information passes through the LGN before reaching the visual cortex. The LGN is organized in 6 distinct layers, each receiving input from one eye. 3 layers receive signals from the eye on the same side, and 3 receive signals from the opposite eye. This layered organization preserves information about which eye detected the visual stimulus, information your brain uses to perceive depth through stereoscopic vision. From the LGN, visual information travels along a pathway called the optic radiations to the primary visual cortex, also known as V1, located in the occipital lobe at the very back of your brain. V1 is the first cortical area dedicated to vision, and it is here that the work of constructing your visual experience truly begins. The neurons in V1 are arranged in columns, each column responding to specific features of the visual stimulus. Some neurons respond preferentially to edges oriented at particular angles, horizontal, vertical, or diagonal. Others respond to direction of motion, firing when something moves across their receptive field in a specific direction. The organization of V1 is remarkably systematic. Adjacent neurons respond to adjacent locations in visual space, creating a map of your visual field on the surface of your cortex. This map is distorted, with the central region of your vision, the fovea, occupying a disproportionately large area of cortex. This magnification reflects the importance of central vision for detailed perception and the high density of photoreceptors in the fovea. When you look directly at an object, you are devoting a substantial portion of your visual cortex to analyzing that small region of space. Visual processing does not stop at V1. Information flows forward to other visual areas, each specialized for different aspects of vision. Area V2 surrounds V1 and continues the analysis of form, color, and motion. Area V4 is particularly involved in color perception and the recognition of shapes. Area V5, also called MT, specializes in detecting motion and predicting the trajectory of moving objects. The inferior temporal cortex is crucial for object recognition, containing neurons that respond selectively to complex shapes like faces. The parietal cortex processes spatial relationships and guides visually directed movements. These different processing streams have been characterized as two main pathways. The ventral stream, sometimes called the Watt pathway, extends from V1 through the temporal lobe and is involved in identifying objects and recognizing patterns. The dorsal stream, called the Where pathway, extends from V1 through the parietal lobe and processes spatial information and motion, guiding actions. Damage to the ventral stream can leave a person able to reach for objects but unable to recognize what they are. Damage to the dorsal stream can impair spatial perception and visually guided movement while leaving object recognition intact. What is extraordinary about visual processing is how much work your brain must do to create the seamless, coherent visual experience you take for granted. The image on your retina is two-dimensional, inverted, and constantly shifting as your eyes move. Yet you perceive a stable three-dimensional world. Your brain integrates information from both eyes to extract depth. It compensates for eye movements so that the world appears stationary, even as the retinal image jumps around. It fills in the blind spot, completes partially occluded objects, and interprets ambiguous patterns based on context and past experience. Your visual system is also constantly making predictions. Rather than passively receiving information, your brain actively generates expectations about what you are likely to see based on what you have seen before. These predictions influence how you interpret incoming sensory data, sometimes causing you to see what you expect rather than what is actually there. This predictive processing allows for faster, more efficient perception, but it can also produce visual illusions when your expectations and reality diverge. The entire pathway from light entering your eye to conscious visual perception takes only a fraction of a second, yet involves billions of neurons working in coordinated patterns. Photons strike photoreceptors. Chemical cascades hyperpolarize cells. Ganglion cells generate action potentials. Signals cross the optic chiasm, pass through the LGN, activate V1, and propagate through higher visual areas. At every stage, the visual signal is transformed, filtered, enhanced, and interpreted. What begins as patterns of light becomes meaning, recognition, awareness. Your eyes are moving right now, even if you are not aware of it. Tiny movements called microsaccades prevent your vision from fading when you stare at a fixed point. Your pupils are adjusting to ambient light levels. Your lenses are making small accommodative changes, and your photoreceptors are continuously transducing light into neural signals, feeding information into the vast processing networks that create your visual world. The structure of your eye is a testament to evolutionary refinement. The eyeball itself is protected by layers of tissue. The outermost layer is the sclera, the tough white coating visible when you look at someone's eye. The sclera provides structural support and protects the delicate internal components. At the front of the eye, the sclera becomes the transparent cornea, allowing light to enter. The middle layer is the choroid, a thin membrane filled with blood vessels that nourishes the retina. The choroid also contains melanin, a dark pigment that absorbs stray light and prevents it from scattering inside the eye, which would blur your vision. The eye is constantly maintaining itself. Tears, produced by lacrimal glands, wash across the surface of your cornea with every blink, keeping it moist and clean. The aqueous humor is continuously produced and drained, maintaining proper pressure inside the eye. If drainage becomes blocked, pressure can build up, damaging the optic nerve in a condition called glaucoma. The retina has the highest metabolic rate of any tissue in your body, consuming oxygen and nutrients at an extraordinary rate to support the constant work of phototransduction. The precision required for clear vision is remarkable. For an image to appear sharp, light from a single point in space must converge on a single point on your retina. This requires the cornea and lens to bend light by exactly the right amount. If the eyeball is slightly too long, light converges before reaching the retina, causing distant objects to appear blurry, a condition called myopia or nearsightedness. If the eyeball is too short, light would converge behind the retina, making nearby objects blurry, hyperopia or farsightedness. Even small deviations from the ideal shape can significantly impact visual clarity. Your two eyes work together to provide you with capabilities that a single eye cannot. Because your eyes are separated by a few centimeters, they see the world from slightly different angles. Your brain compares these two views, and the small differences between them, called binocular disparity, provide information about depth. Objects that are closer to you produce larger disparities. Objects farther away produce smaller disparities. This stereoscopic vision allows you to judge distances with precision, to reach for objects accurately, to navigate through three-dimensional space. But depth perception does not rely solely on binocular disparity. Even with one eye closed, you can estimate distance using monocular cues. Objects that are farther away appear smaller, a phenomenon called perspective. Closer objects partially occlude more distant ones. Parallel lines appear to converge toward the horizon. The sharpness of details decreases with distance due to atmospheric scattering, an effect called aerial perspective. Your brain uses all of these cues simultaneously, integrating them to construct a three-dimensional representation of space from two-dimensional retinal images. Color vision is another sophisticated achievement of your visual system. The ability to distinguish wavelengths of light provides valuable information about objects in your environment. Ripe fruit appears red or yellow against green foliage. A pale face may indicate illness. Color helps you identify objects quickly and discriminate between things that might otherwise look similar. Your perception of color begins with the three types of cones in your retina. But color experience is constructed by your brain through complex comparisons and calculations. The colors you see do not exist in the external world. Light has wavelengths, but wavelength is not color. Color is a perceptual experience created by your nervous system. The same wavelength can appear different in different contexts, and different combinations of wavelengths can produce the same perceived color. Your brain constructs color by comparing the responses of your three cone types and interpreting those relationships within the context of the entire visual scene. This is why colors can appear to shift depending on surrounding colors, a phenomenon artists have exploited for centuries. Vision is also deeply connected to your other senses. What you see influences what you hear. The famous McGurk effect demonstrates this. When you watch someone's lips form one syllable while hearing a different syllable, you often perceive a third syllable that represents a fusion of the visual and auditory information. Your visual system helps you locate sounds in space. When you see something moving, you expect to hear sounds coming from that location, and your brain integrates these expectations into your auditory perception. Movement is especially important in vision. Much of your visual cortex is dedicated to detecting and analyzing motion. This makes evolutionary sense. Moving objects are often more important than stationary ones. A predator in motion requires immediate attention. Food that moves might escape. Your visual system is exquisitely sensitive to motion, capable of detecting movement even in your peripheral vision when you might not notice a stationary object in the same location. This is why a flicker of movement instantly captures your attention, orienting your gaze toward the source. As you lie here now, your visual system continues processing even in darkness. With eyes closed, dim light still penetrates your eyelids, activating photoreceptors. Your brain continues generating visual activity, sometimes producing the faint patterns and colors you might perceive in the darkness. Even in sleep, your visual cortex remains active, contributing to the vivid imagery of dreams. Vision, in some form, never truly stops. The machinery of sight, cornea, lens, retina, optic nerve, cortex, is working continuously, translating light into meaning. But light is only one way the world reaches you. There is another form of energy propagating through the air around you, another set of receptors waiting to detect it, another sensory world to explore. From the realm of photons, we shift now to the realm of vibration, where air itself becomes the carrier of information. Sound does not exist in silence. It requires movement, vibration, the rapid compression and expansion of molecules in the air around you. When an object moves, it pushes against air molecules, creating regions of higher pressure that propagate outward like ripples on water. These pressure waves travel through the air at approximately 343 meters per second, carrying information about their source. When they reach your ears, specialized structures capture these vibrations and transform them into neural signals your brain interprets as sound. The journey begins with the part of your ear you can see, the pinna or auricle. This curved flap of cartilage and skin is not merely decorative. Its complex folds and ridges help collect sound waves and funnel them into your ear canal. The shape of the pinna also provides subtle cues about the direction a sound is coming from, particularly whether it originates above or below you. Sound waves bounce off the pinna's contours in patterns that vary depending on their angle of arrival, and your brain has learned to interpret these patterns. The ear canal, or external auditory meatus, is a tube approximately 2.5 centimeters long that extends from the pinna to the eardrum. The canal is not simply a passive pathway. Its length and shape create resonance at certain frequencies, naturally amplifying sounds in the range most important for understanding human speech, roughly between 2,000 and 5,000 hertz. The canal also contains specialized glands that produce cerumen, commonly known as earwax. Though often considered merely a nuisance, earwax serves important protective functions, trapping dust and debris, preventing infection, and keeping the delicate eardrum from drying out. At the end of the ear canal lies the tympanic membrane, or eardrum. This thin membrane, less than 1/10 of a millimeter thick, stretches across the opening like the head of a drum. When sound waves reach the eardrum, they cause it to vibrate. High-pressure regions push the membrane inward. Low-pressure regions allow it to move outward. The eardrum vibrates in exact correspondence with the incoming sound waves, faithfully reproducing their pattern of compressions and rarefactions. A loud sound creates large movements. A quiet sound produces barely perceptible oscillations. The eardrum marks the boundary between the outer ear and the middle ear, a small air-filled cavity inside your temporal bone. The middle ear contains 3 of the smallest bones in your body, collectively called the ossicles. These bones are named for their shapes: the malleus, hammer; the incus, anvil; and the stapes, stirrup. The malleus is attached to the inner surface of the eardrum. When the eardrum vibrates, the malleus moves with it, and this movement is transmitted through the incus to the stapes. The ossicles do more than simply relay vibrations. They amplify them. The eardrum has a much larger surface area than the oval window, the membrane where the stapes connects to the inner ear. When vibrations are transferred from the larger eardrum to the smaller oval window through the lever action of the ossicles, the pressure is concentrated, increasing by a factor of approximately 20. This amplification is crucial because sound must transition from traveling through air in the outer and middle ear to traveling through fluid in the inner ear. Without amplification, much of the sound energy would simply reflect off the fluid-filled inner ear rather than entering it. The middle ear also contains 2 tiny muscles that can adjust the transmission of sound. The tensor tympani attaches to the malleus. The stapedius attaches to the stapes. When these muscles contract, they stiffen the ossicular chain, reducing the transmission of vibrations. This acoustic reflex occurs automatically in response to loud sounds, providing some protection against noise damage. However, the reflex is too slow to protect against sudden loud noises like gunshots or explosions. The muscles contract within about 40 to 160 milliseconds of a sound's onset, which means the initial, potentially damaging portion of a sudden loud noise reaches the inner ear before protection can engage. The middle ear is connected to the back of your throat by a tube called the eustachian tube. This tube is normally closed but opens when you swallow, yawn, or chew, allowing air to enter or leave the middle ear. This equalization of pressure is essential for proper hearing. The eardrum can vibrate freely only when the air pressure on both sides is equal. This is why your ears feel blocked when you change altitude rapidly, and why swallowing often provides relief. The change in altitude creates a pressure difference across the eardrum until the eustachian tube opens and equalizes the pressure. Beyond the oval window, where the stapes delivers its amplified vibrations, lies the inner ear. This is where mechanical vibrations are finally converted into electrical signals. The inner ear contains 2 separate sensory systems, housed within a labyrinth of fluid-filled chambers carved into your temporal bone. One system, the vestibular apparatus, detects head position and movement. The other, the cochlea, is dedicated to hearing. The cochlea is a spiral structure, coiled like a snail shell, making approximately 2.75 turns. If you could unwind it, the cochlea would be about 35 millimeters long. Inside this spiral is a complex arrangement of 3 fluid-filled chambers separated by membranes. When the stapes pushes against the oval window, it creates pressure waves in the fluid filling the cochlea. These waves travel along the length of the cochlea, causing one of the membranes, the basilar membrane, to move up and down. The basilar membrane is not uniform along its length. At the base of the cochlea, near the oval window, it is narrow and stiff. At the apex, the far end of the spiral, it is wider and more flexible. This gradient in mechanical properties means that different parts of the basilar membrane respond maximally to different frequencies of sound. High-frequency sounds cause maximum movement near the stiff base. Low-frequency sounds cause maximum movement near the flexible apex. This organization creates a frequency map along the length of the cochlea, with each position corresponding to a particular pitch. Scientists call this tonotopic organization. Resting on the basilar membrane is the organ of Corti, the structure that contains the actual sensory receptors for hearing. These receptors are specialized cells called hair cells, and they are among the most remarkable sensory receptors in your body. Each cochlea contains approximately 16,000 hair cells arranged in rows. There are 2 types, a single row of inner hair cells, which do most of the actual sensing, and 3 rows of outer hair cells, which amplify and fine-tune the mechanical response of the cochlea. Extending from the top of each hair cell are tiny projections called stereocilia. These are not true cilia, but rather specialized microvilli, elongated extensions of the cell membrane supported by rigid protein filaments. Each hair cell has between 30 and 300 stereocilia, arranged in rows of increasing height, creating a staircase-like pattern. The stereocilia are the key to hearing. When the basilar membrane moves up and down in response to sound, it causes the stereocilia to bend. The stereocilia are connected to each other by extraordinarily thin protein filaments called tip links. These molecular springs stretch between the top of one stereocilium and the side of the taller stereocilium next to it. When the bundle of stereocilia bends toward the tallest member, the tip links are stretched, pulling open mechanically gated ion channels at their attachment points. This allows potassium and calcium ions to flow into the hair cell. When the stereocilia bend in the opposite direction, the tip links slacken and the channels close. The opening and closing of these channels changes the electrical potential inside the hair cell. When channels open, the influx of positively charged ions depolarizes the cell, making its interior more positive. This depolarization causes voltage-gated calcium channels at the base of the hair cell to open, allowing calcium to enter. The calcium triggers the release of neurotransmitter molecules from the hair cell onto the auditory nerve fibers that contact it. When the stereocilia bend in the opposite direction and the mechanically gated channels close, the cell hyperpolarizes, neurotransmitter release decreases, and the auditory nerve fibers reduce their firing rate. This process happens with extraordinary speed. Hair cells can respond to vibrations occurring thousands of times per second, tracking the waveform of incoming sounds with microsecond precision. This temporal fidelity allows your auditory system to preserve the fine timing information in sounds, which is crucial for understanding speech, localizing sounds, and appreciating music. The speed of hair cell transduction rivals that of any sensory receptor in nature. The inner hair cells, arranged in a single row along the length of the cochlea, are the primary sensory receptors. Each inner hair cell connects to multiple auditory nerve fibers, and each auditory nerve fiber receives input from only a few inner hair cells located at a specific position along the cochlea. This arrangement preserves the tonotopic organization. Each nerve fiber responds most strongly to a particular frequency of sound, determined by where along the cochlea its hair cells are located. Low-frequency sounds activate nerve fibers connected to hair cells near the apex. High-frequency sounds activate fibers connected to hair cells near the base. The outer hair cells, arranged in 3 rows, serve a different function. They act as biological amplifiers. In response to sound, outer hair cells not only receive mechanical input, but also generate mechanical output. They contain a motor protein called prestin embedded in their cell membranes. When the cell's electrical potential changes, prestin changes shape, causing the entire cell to lengthen or shorten. This cellular motion, called electromotility, occurs at the same frequency as the incoming sound. The outer hair cells essentially push back on the structures around them, amplifying the movement of the basilar membrane and sharpening the tuning of the cochlea. This active amplification is what gives your hearing its remarkable sensitivity and frequency discrimination. It allows you to hear sounds so faint that they move the eardrum by less than the diameter of a hydrogen atom. It allows you to distinguish between sounds that differ in frequency by less than 1%. When outer hair cells are damaged, hearing sensitivity decreases dramatically, and sounds become muffled and difficult to distinguish. This is the most common cause of noise-induced and age-related hearing loss. The auditory nerve fibers that receive signals from the inner hair cells bundle together to form the auditory nerve, also called the cochlear nerve. This nerve exits the cochlea and travels to the brainstem. Unlike visual information, which crosses over at the optic chiasm, auditory information from each ear is processed by both sides of the brain. This bilateral processing begins immediately at the first relay station in the brainstem, the cochlear nucleus. From there, signals follow multiple parallel pathways through the brainstem and midbrain. One important brainstem structure for hearing is the superior olivary complex. This is where your brain begins to extract information about the location of sounds. Sound coming from your right side reaches your right ear slightly before reaching your left ear. The difference might be only a few hundred microseconds, but your auditory system can detect it. The superior olivary complex contains neurons that compare the timing and intensity of signals arriving from the two ears, using these tiny differences to determine where a sound is coming from. As auditory information ascends through the brainstem, it maintains its tonotopic organization. Each processing station contains a frequency map, with neurons responding to low frequencies grouped in one region and neurons responding to high frequencies grouped in another. This organization is preserved all the way to the auditory cortex. Eventually, auditory signals reach the thalamus, specifically a region called the medial geniculate nucleus, which serves as the final relay before information reaches the cerebral cortex. From the medial geniculate nucleus, auditory information projects to the primary auditory cortex, located in the temporal lobe of your brain. Here, in a region called A1, neurons are arranged tonotopically, just as they are at every earlier stage of processing. Neurons that respond to low-frequency sounds are located in one area. Neurons that respond to high-frequency sounds are in another, creating a map of frequency across the cortical surface. But the auditory cortex does more than simply represent frequency. Neurons in this region respond to complex features of sound, temporal patterns, amplitude modulations, spectral combinations. Some neurons respond selectively to specific speech sounds or environmental noises. Through this hierarchical processing, the vibrations that began in the air around you are transformed into meaningful auditory experiences. Right now, even in relative quiet, your auditory system is active. Your hair cells are moving in response to ambient sounds, the subtle hum of ventilation, the distant murmur of traffic, the quiet sounds of your own breathing. Your auditory nerve is carrying a constant stream of information to your brain, and somewhere in the intricate networks of your auditory cortex, this information is being analyzed, categorized, and integrated with information from your other senses. Your ability to locate sounds in space depends on subtle differences in what your two ears detect. When a sound originates to your right, it reaches your right ear first, and with slightly greater intensity, because your head partially blocks the sound from reaching your left ear. For low-frequency sounds, your brain primarily uses the time difference between the two ears. For high-frequency sounds, it relies more on intensity differences. These interaural time and intensity differences are computed by specialized neurons in the superior olivary complex and other brainstem nuclei, allowing you to determine the horizontal direction of a sound source with remarkable precision. Determining whether a sound comes from above or below, or from in front or behind, is more challenging because these locations produce no consistent differences between the two ears. Instead, your brain uses the spectral filtering created by the shape of your pinny. Sounds from different vertical and front-back locations reflect off the folds of your outer ear differently, creating subtle changes in the frequency content that reaches your eardrum. Your brain has learned these patterns through experience and uses them to disambiguate sound source locations. This is why sounds sometimes seem to come from unexpected directions when you wear headphones that bypass the natural filtering of your pinny. The perception of loudness is related to sound intensity, but the relationship is not simple. Your auditory system can respond to an enormous range of sound intensities, from the threshold of hearing to sounds more than 1,000,000 times more intense. To handle this range, your perception of loudness follows a logarithmic scale. A sound must increase in intensity by a factor of 10 to be perceived as twice as loud. This compression allows you to function in both quiet libraries and noisy restaurants without being overwhelmed. Pitch. The perceptual quality that makes a note sound high or low is primarily determined by frequency, but your perception of pitch also depends on intensity, duration, and the presence of other sounds. The same frequency can sound slightly different depending on how loud it is. Very brief sounds may not have a clear pitch at all, and when multiple frequencies are present simultaneously, you may perceive a pitch corresponding to a frequency that is not physically present in the sound, a phenomenon called the missing fundamental. Your brain infers the fundamental frequency based on the pattern of harmonics it detects. Timbre is the quality that allows you to distinguish between different instruments playing the same note or different voices speaking the same word. Timbre depends on the spectral content of a sound, the distribution of energy across different frequencies. A violin and a flute playing the same pitch produce sound waves at the same fundamental frequency, but they also produce different patterns of overtones at higher frequencies. Your auditory system analyzes these spectral differences, allowing you to identify sound sources even when pitch and loudness are matched. Speech perception is one of the most complex tasks your auditory system performs. Understanding spoken language requires extracting rapid sequences of acoustic features, segmenting the continuous stream of sound into discrete units, and mapping those units onto learned linguistic categories. Your brain accomplishes this by analyzing multiple acoustic cues simultaneously: the frequency patterns of vowels, the brief silences and bursts of consonants, the rising and falling intonation that conveys emotion and meaning. The auditory cortex works closely with language areas in the frontal and temporal lobes to transform acoustic patterns into words, sentences, and ideas. Your auditory system integrates with your other senses to create a unified perceptual experience. What you see influences what you hear. When you watch someone speak, your brain combines the auditory information from their voice with the visual information from their lip movements. This multisensory integration is so powerful that visual information can change your perception of what sounds are being spoken, as demonstrated by the McGurk effect. Seeing lips form one sound while hearing a different sound can cause you to perceive a third sound that represents a blend of the two. Touch also interacts with hearing. When you feel a tap on your body at the same time as you hear a sound, your brain binds these sensations together, enhancing the perception of both. This is why movies feel more immersive when the theater seats vibrate during explosions, or why feeling your phone buzz while hearing it ring creates a stronger sense of an incoming call. The brain regions that process hearing and touch exchange information continuously, allowing for this multisensory binding. The vulnerability of your auditory system becomes apparent when you consider the hair cells in your cochlea. Unlike many cells in your body, hair cells in adult humans do not regenerate when damaged or destroyed. Exposure to loud sounds can cause mechanical damage to the stereocilia or kill the hair cells outright. Once lost, these cells are gone permanently, and with them goes a portion of your hearing. This is why noise-induced hearing loss is irreversible. Protecting your hearing requires limiting exposure to loud sounds or wearing ear protection when loud sounds cannot be avoided. Age also affects hearing. As you grow older, hair cells gradually die, typically beginning with those at the base of the cochlea that respond to high frequencies. This age-related hearing loss, called presbycusis, is nearly universal in older adults. High-pitched sounds become harder to hear, and speech understanding deteriorates, especially in noisy environments. The loss is gradual enough that many people do not notice it until it becomes significant. The journey from vibration to perception is one of extraordinary precision and complexity. Air molecules compress and expand, moving your eardrum by distances smaller than an atom. Tiny bones amplify and transmit these movements. Fluid waves ripple through a spiral of membrane and tissue. Stereocilia bend, ion channels open, neurotransmitters release, and nerve fibers fire in precise patterns. Signals ascend through brainstem, midbrain, thalamus, and cortex. And from this cascade of mechanical and electrical events emerges your experience of sound, a voice, a melody, the rustle of wind. But sound is not the only vibration your body detects. Your skin responds to pressure, movement, and texture through its own population of mechanoreceptors. From the waves propagating through air, we turn now to the physical contact between your body and the world. Your skin is the largest organ in your body, covering approximately 2 square meters and weighing about 4 kilograms. But it is far more than a protective barrier. Your skin is a vast sensory surface, densely populated with millions of receptors that continuously monitor your physical contact with the world. Every texture you feel, every brush of air, every embrace, all of these experiences begin with specialized cells embedded in the layers of your skin. Touch is different from your other senses in a fundamental way. Vision and hearing have dedicated organs, eyes and ears, concentrated in specific locations on your body. But touch receptors are distributed across your entire skin surface, from the top of your head to the soles of your feet. This distribution means that touch provides information not just about external objects, but also about your body's relationship to the environment at every point of contact. You are always touching something, even if only the air around you or the surface beneath you. The skin itself consists of multiple layers. The outermost layer, the epidermis, is composed primarily of dead cells that provide protection against injury and infection. Beneath this lies the dermis, a thicker layer containing blood vessels, hair follicles, sweat glands, and the majority of your touch receptors. Below the dermis is the subcutaneous layer, consisting of fat and connective tissue that cushions your body and insulates against temperature changes. Touch receptors are embedded at different depths within these layers, each positioned to detect specific types of mechanical stimulation. There are 4 main types of mechanoreceptors in your skin, each with a unique structure and function. Merkel shells, also called Merkel discs, are located in the uppermost layer of the dermis, close to the epidermis. These receptors respond to sustained pressure and are exquisitely sensitive to fine details and edges. When you run your fingertip across a textured surface, Merkel cells detect the subtle variations in pressure as the ridges of your fingerprint encounter bumps and indentations. These receptors adapt slowly, meaning they continue firing as long as pressure is applied, providing continuous information about what you are touching. Meissner corpuscles are found in the dermal papillae, small projections of dermis that extend up into the epidermis. These receptors are particularly abundant in areas of your body that require high tactile sensitivity, such as your fingertips, lips, and the soles of your feet. Meissner corpuscles respond to light touch and changes in texture. They adapt rapidly, firing a burst of action potentials when a stimulus first appears or changes, but quickly reducing their response if the stimulus remains constant. This rapid adaptation makes them ideal for detecting movement across the skin, such as when an insect crawls on your arm or when you brush against something. Deeper in the dermis and subcutaneous tissue lie Pacinian corpuscles, the largest of the touch receptors. These onion-shaped structures consist of a nerve ending surrounded by concentric layers of connective tissue. Pacinian corpuscles detect deep pressure and high-frequency vibrations. When you feel the buzz of your phone in your pocket or sense the vibration of machinery through your feet, Pacinian corpuscles are responsible. Like Meissner corpuscles, Pacinian corpuscles adapt rapidly, responding most strongly to changes in pressure rather than sustained force. Ruffini corpuscles, also called Ruffini endings, are elongated receptors located deep in the dermis and in joint capsules. These receptors respond to skin stretch, providing information about the position and movement of your fingers and limbs. When you flex your fingers or rotate your wrist, Ruffini corpuscles detect the stretching of skin and connective tissue. They adapt slowly, continuing to fire as long as the stretch is maintained, which provides ongoing information about limb position. In addition to these 4 main types of mechanoreceptors, your skin contains receptors for temperature and pain. Thermoreceptors detect warmth and cold through specialized ion channels that open at specific temperatures. Free nerve endings, which are simply unmyelinated nerve fibers that terminate in the skin without specialized structures, detect painful stimuli, intense pressure, and temperature extremes. These nociceptors serve a protective function, alerting you to potentially harmful conditions before serious damage occurs. The density of touch receptors varies dramatically across different regions of your body. Your fingertips have the highest concentration of receptors, particularly Merkel cells and Meissner corpuscles. giving them extraordinary sensitivity. You can detect features smaller than a millimeter with your fingertips, distinguishing textures and identifying objects through touch alone. Your lips and tongue are similarly well equipped with touch receptors, reflecting their importance for eating, speaking, and facial expression. In contrast, the skin on your back has a much lower receptor density, which is why 2 points touching your back must be several centimeters apart before you can perceive them as separate. This variation in receptor density creates a distorted map of your body's sensitivity. If you were to draw your body with each part sized according to its tactile sensitivity rather than its physical size, you would have enormous hands, lips, and face with a relatively small torso and limbs. This distortion is preserved in your brain, where the somatosensory cortex devotes disproportionate space to processing information from your most sensitive areas. When a mechanoreceptor in your skin is stimulated, it generates an electrical signal that travels along a sensory neuron toward your spinal cord. These neurons are part of the peripheral nervous system, with their cell bodies located in clusters, called dorsal root ganglia just outside the spinal cord. The neurons vary in diameter and myelination, which affects how quickly they conduct signals. Large, heavily myelinated fibers carry information about touch and vibration rapidly, at speeds up to 120 meters per second. Smaller, lightly myelinated, or unmyelinated fibers carry information about temperature and pain more slowly. When touch and proprioceptive signals from your body reach the spinal cord, they enter through the dorsal roots, bundles of nerve fibers at the back of each spinal segment. Once inside the spinal cord, these signals follow one of two main pathways to the brain. Information about fine touch, vibration, and proprioception ascends through the dorsal column medial lemniscus pathway. This pathway gets its name from the dorsal columns, two bundles of nerve fibers that run up the back of the spinal cord, and the medial lemniscus, the tract these fibers form after crossing over in the brainstem. The dorsal columns maintain strict organization. Fibers entering from lower parts of your body, carrying information from your legs and lower trunk, travel in the medial portion of the dorsal columns. Fibers from higher segments, carrying information from your arms and upper body, are added laterally as the pathway ascends. This arrangement preserves a map of your body within the spinal cord itself. The fibers in the dorsal columns travel all the way up to the medulla, the lowest part of your brainstem, without crossing to the opposite side. In the medulla, the dorsal column fibers synapse onto neurons in two nuclei. The gracile nucleus, which receives input from the lower body, and the cunate nucleus, which receives input from the upper body. From these nuclei, second-order neurons send their axons across the midline, crossing to the opposite side of the brainstem. This crossing, called decussation, means that touch information from the left side of your body is processed by the right side of your brain, and vice versa. After crossing, the fibers form the medial lemniscus, which continues upward through the brainstem to the thalamus. The thalamus, serving as the brain's sensory relay station, receives touch information in a region called the ventral posterior nucleus. This nucleus is organized somatotopically, meaning it contains a map of your body surface. Neurons responding to touch on your foot are located in one area, neurons responding to your hand are in another. The thalamus does not simply relay information passively, but modulates and filters it, influenced by attention, arousal, and input from the cortex. From the thalamus, third-order neurons project to the primary somatosensory cortex in the parietal lobe. The primary somatosensory cortex, designated S1, occupies a strip of tissue running along the post-central gyrus, just behind the central sulcus, that divides the frontal and parietal lobes. Like every structure before it in the pathway, S1 is organized somatotopically. The organization creates what is called the sensory homunculus. a distorted map of the human body laid out across the cortical surface. The representation is inverted, with your feet mapped to the top of the cortex near the midline, your trunk and arms along the side, and your face and lips at the bottom. The size of each body part's representation corresponds to its sensory importance rather than its physical size. Your hands, lips, and tongue occupy enormous areas of cortex. Your back and legs occupy relatively small areas. Within S1, there are distinct subregions that process different aspects of touch. One area receives input primarily from receptors that respond to texture. Another processes information about vibration. Still another is involved in detecting the shape and contours of objects. These subregions work in parallel, each extracting particular features from the tactile input, then sharing information with each other to build a complete representation of what you are touching. Beyond S1 lies the secondary somatosensory cortex, S2, located in a region where the parietal lobe curves down toward the temporal lobe. S2 receives input from S1 and performs higher-level processing, integrating information from both sides of your body. While S1 neurons typically respond to touch on only one side of your body, S2 neurons often respond to stimulation of either side. This bilateral integration is important for tasks that require coordinating both hands, such as tying shoelaces or manipulating objects. The somatosensory cortex does not work in isolation. It exchanges information extensively with the motor cortex, which controls movement. This connection is essential because effective action requires knowing where your body parts are and what they are touching. When you reach for an object, your somatosensory cortex provides continuous feedback about contact and grip force, allowing your motor cortex to adjust the movement in real time. Damage to the somatosensory cortex impairs not only sensation but also the ability to perform skilled movements, even though the motor cortex itself remains intact. Touch also interacts extensively with other sensory systems. What you see influences what you feel. If you watch someone stroke a fake hand while your real hand is stroked in synchrony, you may begin to feel as though the touch is coming from the fake hand, the rubber hand illusion. This illusion demonstrates how powerfully vision can influence tactile perception. Your brain integrates visual and tactile information to determine where your body is and what is touching it. And when these two sources of information are consistent, they reinforce each other. Sound and touch also interact, particularly for detecting vibrations. When you feel vibration and hear a corresponding sound simultaneously, your perception of both is enhanced. This is why turning up the bass in music makes you feel the rhythm more intensely, even if you are not directly touching the speakers. The sensory cortices for hearing and touch are anatomically close and share connections, allowing for this cross-modal interaction. Touch is essential for emotional connection and social bonding. The sensation of being touched by another person activates not only the somatosensory cortex, but also brain regions involved in emotion and social cognition. A gentle touch can be comforting, reassuring, calming. Research has shown that slow, gentle stroking of the skin activates a special class of nerve fibers called C-tactile afferents, which project to emotional rather than sensory areas of the brain. These fibers may be specialized for detecting the kind of touch that occurs during social interactions, contributing to the emotional quality of physical contact. Your sense of touch is constantly active, providing a continuous stream of information about your body's interaction with the environment. Right now, receptors in your skin are responding to pressure, temperature, and texture. Signals are traveling up your spinal cord, crossing in your brainstem, relaying through your thalamus, and activating your somatosensory cortex. This information is being integrated with visual and auditory input, combined with motor commands, and contributing to your sense of your body in space. Touch tells you where the world ends and you begin. But there are sensations that do not come from external contact, internal experiences that arise from within your mouth and nose, chemical signals that become flavor. The ability to distinguish between two separate points of contact on your skin depends on receptor density and neural processing. This capability, called two-point discrimination, varies dramatically across your body. On your fingertips, you can feel 2 points as separate when they are only 2 to 3 millimeters apart. On your forearm, the points must be separated by about 4 centimeters before you perceive them as distinct. On your back, the threshold increases to 7 centimeters or more. This variation reflects both the density of receptors in different regions and the amount of cortical space devoted to processing information from those regions. Your brain's interpretation of touch is not simply a passive recording of receptor activity. It is an active construction that can be influenced by context, expectation, and input from other senses. Tactile illusions demonstrate this clearly. If you cross your fingers and touch your nose, it may momentarily feel as though you are touching two noses. Your brain expects that sensations on opposite sides of two different fingers come from two different objects, and when this expectation conflicts with reality, perception becomes confused. Another striking tactile illusion occurs when you hold a small object between crossed fingers and roll it back and forth. The object may feel as though it splits into two separate objects. This happens because your brain interprets the pattern of stimulation, touch on the outer edge of one finger and the outer edge of another, as arising from two distinct objects, rather than a single object bridging the gap. These illusions reveal that touch, like all perception, is not a direct readout of sensory input, but an inference your brain constructs based on how stimuli are most likely to be arranged in the world. Temperature, sensation is closely related to touch, but involves different receptors. Your skin contains thermoreceptors that respond to warming or cooling. Cold receptors increase their firing rate when temperature decreases. Warm receptors respond to temperature increases. These receptors contain specialized ion channels called TRP channels, which open at specific temperatures. Different TRP channels have different activation thresholds, allowing your nervous system to distinguish between cool, neutral, warm, and hot temperatures. Interestingly, both extreme cold and extreme heat can activate the same pain receptors. This is why very cold objects can feel burning hot, and why your brain sometimes has difficulty distinguishing between extreme temperatures. The sensation of cold pain and heat pain both travel along similar neural pathways to the brain, and at extreme intensities, the distinction between them becomes blurred. The perception of wetness is particularly intriguing because there are no dedicated wetness receptors in your skin. Instead, your brain infers wetness from a combination of temperature and touch information. Cool temperature combined with light, slippery touch tends to be interpreted as wetness. This is why a cold, smooth object can sometimes fool you into thinking your hand is wet when it is actually dry. Your perception of wetness is a constructed interpretation based on patterns of activity across multiple receptor types. Pain, though unpleasant, serves a crucial protective function. Nociceptors in your skin detect stimuli that are potentially harmful. Intense pressure, extreme temperatures, tissue damage, and inflammatory chemicals. When activated, these receptors send urgent signals through the nervous system, often triggering reflexes that pull your body away from danger before you are even consciously aware of the threat. A hot stove activates nociceptors in your fingertip. Signals race to your spinal cord, and your hand jerks back, all within a fraction of a second, before the signal has time to reach your brain and become a conscious sensation. The pathways that carry pain and temperature information differ from those that carry touch information. Rather than ascending through the dorsal columns, pain and temperature signals cross to the opposite side of the spinal cord almost immediately after entering, and then travel upward through a pathway called the spinothalamic tract. This pathway projects to the thalamus and then to multiple cortical areas, including the somatosensory cortex and regions involved in emotion and attention. This explains why pain has both a sensory component, the location and intensity of the stimulus, and an emotional component, the unpleasantness and urgency that motivate you to respond. Adaptation plays a crucial role in touch perception. As mentioned earlier, some receptors adapt quickly, responding strongly to changes, but quickly reducing their firing if stimulation remains constant. This is why you stop feeling your clothes against your skin shortly after getting dressed. The receptors detected the pressure when the fabric first made contact, but they adapted, allowing your attention to focus on new or changing tactile information rather than being distracted by constant, unchanging stimulation. Other receptors adapt slowly, continuing to fire as long as pressure is maintained, which is important for tasks requiring sustained grip or for detecting objects that remain in contact with your skin. Your tactile system maintains its sensitivity through complex feedback mechanisms. When you actively explore an object with your hands, your brain adjusts the sensitivity of your touch receptors based on what you are trying to detect. If you are searching for fine texture, activity in your somatosensory cortex enhances signals from receptors that respond to high-frequency vibration. If you are assessing firmness, different receptors and cortical areas are emphasized. This attentional modulation allows you to extract different information from the same object depending on your goals. Touch is fundamentally about contact, about the physical interface between your body and everything else. It tells you about surfaces and textures, temperatures and pressures, pain and pleasure. It provides the immediate visceral sense that you are a physical being in a physical world. Every moment, your skin is gathering information, your neurons are transmitting signals, your brain is constructing tactile experience, but not all sensing requires physical contact. There are molecules drifting through the air and dissolved in your saliva, chemical messengers that reach receptors in your nose and mouth. These are the signals that become taste and smell, senses that detect the molecular composition of your world. While light and sound carry information through physical waves, and touch relies on mechanical contact, there is another way the world reaches you. Through molecules. Every object releases chemical compounds into the air or dissolves substances when placed in liquid. Your body has evolved sensory systems specifically designed to detect these molecules, to analyze their chemical structure, and to transform that information into the experiences you call taste and smell. These chemical senses are ancient, predating vision and hearing in evolutionary history, and they serve fundamental functions: identifying food, detecting danger, and triggering powerful memories and emotions. Taste and smell are separate sensory systems with different receptors, different neural pathways, and different regions of the brain dedicated to processing their signals. Yet they work together so intimately that you experience them as a unified sensation. What you commonly refer to as the taste of food is actually a combination of true taste detected by your tongue, smell detected by your nose, texture sensed by mechanoreceptors in your mouth, and even temperature and pain detected by thermoreceptors and nociceptors. When you eat, all of these sensory inputs converge, creating the rich, complex experience called flavor. True taste, the sensation detected by your tongue, is remarkably simple compared to smell. Your taste system can distinguish only 5 basic qualities: sweet, salty, sour, bitter, and umami. Sweet taste indicates the presence of sugars, an important energy source. Salty taste reflects the concentration of sodium and other minerals essential for bodily functions. Sour taste signals acidity, which can indicate unripe or spoiled food. Bitter taste warns of potentially toxic compounds, many of which taste intensely bitter even at very low concentrations. Umami, a Japanese word meaning delicious savory taste, responds to amino acids, particularly glutamate, signaling the presence of protein. The receptors for taste are located in taste buds, small clusters of specialized cells embedded in the surface of your tongue, soft palate, throat, and upper esophagus. Most taste buds reside on your tongue, particularly in small bumps called papillae that you can see and feel on your tongue's surface. Each taste bud contains 50 to 100 taste receptor cells arranged like segments of an orange. These cells are not neurons but specialized epithelial cells that detect chemical stimuli and transmit signals to sensory nerve fibers. Taste receptor cells have microvilli extending from their apical surface into a small opening called the taste pore. When you eat or drink, dissolved molecules enter the taste pore and interact with receptors on the microvilli. Different taste qualities are detected through different molecular mechanisms. Salty and sour tastes are detected by ion channels that allow sodium or hydrogen ions to enter the taste cell directly. When these ions flow in, they depolarize the cell, triggering the release of neurotransmitter onto the sensory nerve fibers at the base of the taste cell. Sweet, bitter, and umami tastes are detected by G-protein coupled receptors, specialized proteins embedded in the cell membrane. When a molecule with the appropriate chemical structure binds to one of these receptors, it activates a signaling cascade inside the cell. This cascade ultimately leads to the release of calcium from internal stores, which depolarizes the cell and triggers neurotransmitter release. The G-protein coupled receptors for taste are remarkably selective. Sweet receptors respond to various sugars and some artificial sweeteners. Umami receptors bind glutamate and related amino acids. Bitter receptors are more diverse, with about 25 different types that collectively detect a wide range of potentially harmful compounds. Another striking tactile illusion occurs when you hold a small object between crossed fingers and roll it back and forth. The object may feel as though it splits into two separate objects. This happens because your brain interprets the pattern of stimulation, touch on the outer edge of one finger and the outer edge of another, as arising from two distinct objects, rather than a single object bridging the gap. These illusions reveal that touch, like all perception, is not a direct readout of sensory input, but an inference your brain constructs based on how stimuli are most likely to be arranged in the world. Temperature, sensation is closely related to touch, but involves different receptors. Your skin contains thermoreceptors that respond to warming or cooling. Cold receptors increase their firing rate when temperature decreases. Warm receptors respond to temperature increases. These receptors contain specialized ion channels called TRP channels, which open at specific temperatures. Different TRP channels have different activation thresholds, allowing your nervous system to distinguish between cool, neutral, warm, and hot temperatures. Interestingly, both extreme cold and extreme heat can activate the same pain receptors. This is why very cold objects can feel burning hot, and why your brain sometimes has difficulty distinguishing between extreme temperatures. The sensation of cold pain and heat pain both travel along similar neural pathways to the brain and at extreme intensities. The distinction between them becomes blurred. The perception of wetness is particularly intriguing because there are no dedicated wetness receptors in your skin. Instead, your brain infers wetness from a combination of temperature and touch information. Cool temperature combined with light, slippery touch tends to be interpreted as wetness. This is why a cold, smooth object can sometimes fool you into thinking your hand is wet when it is actually dry. Your perception of wetness is a constructed interpretation based on patterns of activity across multiple receptor types. Pain, though unpleasant, serves a crucial protective function. Nociceptors in your skin detect stimuli that are potentially harmful. Intense pressure, extreme temperatures, tissue damage, and inflammatory chemicals. When activated, these receptors send urgent signals through the nervous system, often triggering reflexes that pull your body away from danger before you are even consciously aware of the threat. A hot stove activates nociceptors in your fingertip. Signals race to your spinal cord, and your hand jerks back, all within a fraction of a second, before the signal has time to reach your brain and become a conscious sensation. The pathways that carry pain and temperature information differ from those that carry touch information. Rather than ascending through the dorsal columns, pain and temperature signals cross to the opposite side of the spinal cord almost immediately after entering, and then travel upward through a pathway called the spinothalamic tract. This pathway projects to the thalamus and then to multiple cortical areas, including the somatosensory cortex and regions involved in emotion and attention. This explains why pain has both a sensory component, the location and intensity of the stimulus, and an emotional component, the unpleasantness and urgency that motivate you to respond. Adaptation plays a crucial role in touch perception. As mentioned earlier, some receptors adapt quickly, responding strongly to changes, but quickly reducing their firing if stimulation remains constant. This is why you stop feeling your clothes against your skin shortly after getting dressed. The receptors detected the pressure when the fabric first made contact, but they adapted, allowing your attention to focus on new or changing tactile information rather than being distracted by constant, unchanging stimulation. Other receptors adapt slowly, continuing to fire as long as pressure is maintained, which is important for tasks requiring sustained grip or for detecting objects that remain in contact with your skin. Your tactile system maintains its sensitivity through complex feedback mechanisms. When you actively explore an object with your hands, your brain adjusts the sensitivity of your touch receptors based on what you are trying to detect. If you are searching for fine texture, activity in your somatosensory cortex enhances signals from receptors that respond to high-frequency vibration. If you are assessing firmness, different receptors and cortical areas are emphasized. This attentional modulation allows you to extract different information from the same object depending on your goals. Touch is fundamentally about contact, about the physical interface between your body and everything else. It tells you about surfaces and textures, temperatures and pressures, pain and pleasure. It provides the immediate visceral sense that you are a physical being in a physical world. Every moment, your skin is gathering information, your neurons are transmitting signals, your brain is constructing tactile experience, but not all sensing requires physical contact. There are molecules drifting through the air and dissolved in your saliva, chemical messengers that reach receptors in your nose and mouth. These are the signals that become taste and smell, senses that detect the molecular composition of your world. While light and sound carry information through physical waves, and touch relies on mechanical contact, there is another way the world reaches you. Through molecules. Every object releases chemical compounds into the air or dissolves substances when placed in liquid. Your body has evolved sensory systems specifically designed to detect these molecules, to analyze their chemical structure, and to transform that information into the experiences you call taste and smell. These chemical senses are ancient, predating vision and hearing in evolutionary history, and they serve fundamental functions: identifying food, detecting danger, and triggering powerful memories and emotions. Taste and smell are separate sensory systems with different receptors, different neural pathways, and different regions of the brain dedicated to processing their signals. Yet they work together so intimately that you experience them as a unified sensation. What you commonly refer to as the taste of food is actually a combination of true taste detected by your tongue, smell detected by your nose, texture sensed by mechanoreceptors in your mouth, and even temperature and pain detected by thermoreceptors and nociceptors. When you eat, all of these sensory inputs converge, creating the rich, complex experience called flavor. True taste, the sensation detected by your tongue, is remarkably simple compared to smell. Your taste system can distinguish only 5 basic qualities: sweet, salty, sour, bitter, and umami. Sweet taste indicates the presence of sugars, an important energy source. Salty taste reflects the concentration of sodium and other minerals essential for bodily functions. Sour taste signals acidity, which can indicate unripe or spoiled food. Bitter taste warns of potentially toxic compounds, many of which taste intensely bitter even at very low concentrations. Umami, a Japanese word meaning delicious savory taste, responds to amino acids, particularly glutamate, signaling the presence of protein. The receptors for taste are located in taste buds, small clusters of specialized cells embedded in the surface of your tongue, soft palate, throat, and upper esophagus. Most taste buds reside on your tongue, particularly in small bumps called papillae that you can see and feel on your tongue's surface. Each taste bud contains 50 to 100 taste receptor cells arranged like segments of an orange. These cells are not neurons but specialized epithelial cells that detect chemical stimuli and transmit signals to sensory nerve fibers. Taste receptor cells have microvilli extending from their apical surface into a small opening called the taste pore. When you eat or drink, dissolved molecules enter the taste pore and interact with receptors on the microvilli. Different taste qualities are detected through different molecular mechanisms. Salty and sour tastes are detected by ion channels that allow sodium or hydrogen ions to enter the taste cell directly. When these ions flow in, they depolarize the cell, triggering the release of neurotransmitter onto the sensory nerve fibers at the base of the taste cell. Sweet, bitter, and umami tastes are detected by G-protein coupled receptors, specialized proteins embedded in the cell membrane. When a molecule with the appropriate chemical structure binds to one of these receptors, it activates a signaling cascade inside the cell. This cascade ultimately leads to the release of calcium from internal stores, which depolarizes the cell and triggers neurotransmitter release. The G-protein coupled receptors for taste are remarkably selective. Sweet receptors respond to various sugars and some artificial sweeteners. Umami receptors bind glutamate and related amino acids. Bitter receptors are more diverse, with about 25 different types that collectively detect a wide range of potentially harmful compounds. Molecules are drifting into your nose right now, activating patterns of receptors, sending signals to your olfactory bulb and cortex. Your tongue carries information about basic tastes, your nose about complex smells, and your brain binds these together into flavor. But there is more to sensing than detecting the external world. Your body also monitors itself, tracking where your limbs are without looking, maintaining a continuous awareness of your position in space. The perception of taste intensity is not simply proportional to the concentration of molecules. Your taste system compresses the range of intensities you can detect, similar to how your visual and auditory systems handle wide ranges of stimulus intensity. Doubling the concentration of sugar in a solution does not make it taste twice as sweet. Instead, perceived intensity increases more slowly than actual concentration, following a logarithmic relationship. This compression allows you to appreciate subtle differences at low concentrations while preventing saturation at high concentrations. Taste adaptation occurs when you are exposed to a taste stimulus for an extended period. The receptors become less responsive, and the perceived intensity diminishes. This is why the first sip of a sweet drink tastes more intensely sweet than subsequent sips. Adaptation serves to highlight changes in your sensory environment rather than maintaining constant awareness of unchanging stimuli. After adaptation to one taste, your sensitivity to other tastes can change. Drinking orange juice after brushing your teeth tastes unpleasant because toothpaste contains compounds that suppress sweet receptors and enhance bitter receptors, dramatically altering your taste perception. The sense of taste undergoes changes throughout your lifespan. Infants are born with more taste buds than adults, distributed not just on the tongue, but throughout the mouth. Children are generally more sensitive to bitter tastes than adults, which may serve as a protective mechanism against poisoning during a vulnerable period of development. As you age, the number of taste buds decreases, and those that remain become less sensitive. This decline in taste sensitivity is one reason why older adults often find food less appealing and may add more salt or sugar to achieve the same flavor intensity. Smell also changes with age, typically declining more dramatically than taste. By age 60, roughly half of people experience some degree of smell loss. This decline can affect quality of life, reducing appetite, impairing the ability to detect spoiled food or dangerous odors like gas leaks. and eliminating one of life's simple pleasures. The loss of smell is also an early indicator of certain neurodegenerative diseases, including Parkinson's disease and Alzheimer's disease. Olfactory testing is increasingly being explored as a potential diagnostic tool for detecting these conditions before other symptoms appear. Cultural and individual differences in taste and smell perception are substantial. What smells pleasant in one culture may smell unpleasant in another, largely due to learned associations. Foods that taste delicious to people accustomed to them may taste strange or unpleasant to those encountering them for the first time. These differences are not merely cultural, but also have biological components. Genetic variations affect not only receptor sensitivity, but also the metabolic enzymes that break down odorants, which can alter how long a smell persists and how it is perceived. The integration of the chemical senses with other sensory modalities creates phenomena that extend beyond simple taste and smell. The color of food influences its perceived flavor. A drink that looks red is expected to taste like cherry or strawberry, and if it actually tastes like lime, the mismatch creates a confusing and often unpleasant experience. The sound of food, the crunch of a chip, the sizzle of a steak, enhances flavor perception. Even the weight and texture of utensils can influence how food tastes. Your brain constructs flavor from a symphony of sensory inputs, with taste and smell as the lead instruments, but with supporting contributions from vision, hearing, and touch. Temperature affects taste perception in complex ways. Cold temperatures generally suppress sweet taste, which is why ice cream manufacturers use high sugar concentrations to achieve the desired sweetness when frozen. Bitter taste is enhanced at higher temperatures, which is why cold coffee tastes less bitter than hot coffee with the same concentration of caffeine and other bitter compounds. These temperature effects reflect the biophysics of receptor activation. Receptor proteins function differently at different temperatures. Pain and irritation also contribute to flavor. The burn of chili peppers comes from capsaicin, a molecule that activates pain receptors in your mouth. The cooling sensation of menthol activates cold receptors. These sensations are not taste or smell, but trigeminal sensations, detected by nerve endings of the trigeminal nerve. Yet they are integrated into your experience of flavor, adding dimensions of heat, coolness, tingling, or burning that enhance or define certain foods. The chemical senses connect you to your environment in intimate ways. They allow you to sample the molecular composition of the world, to detect nutrients and toxins, to experience pleasure and disgust. Every breath brings new molecules to your olfactory epithelium. Every bite dissolves compounds onto your taste buds. These signals travel through cranial nerves to your brainstem, thalamus and cortex, where they are analyzed, integrated, and interpreted. Right now, your chemical senses are active, monitoring the air and your mouth, sending streams of information to your brain. But there are other senses operating continuously. Senses you rarely think about, but could not function without. Senses that tell you where your body is in space, how your limbs are positioned, whether you are moving or still. From the chemistry of molecules, we turn now to the physics of your body in space. Close your eyes and touch your nose with your finger. You can do this easily, without hesitation, without needing to look. Now hold your arms out to the sides and bring your fingertips together behind your back. Again, you know exactly where your hands are, where your arms are positioned, how your joints are angled. This ability to sense the position and movement of your body parts without visual guidance is called proprioception, and it is one of the most fundamental yet least appreciated of your senses. Proprioception is often called the sixth sense, though this term understates its importance. Without proprioception, even the simplest movements become nearly impossible. Walking requires knowing where your feet are in relation to the ground. Grasping an object requires knowing the position of your fingers and the force of your grip. Speaking requires precise control of your tongue, lips, and jaw, all of which depends on proprioceptive feedback. Every voluntary movement your body makes relies on a continuous stream of information from proprioceptive receptors embedded in your muscles, tendons, and joints. These receptors detect mechanical changes in your musculoskeletal system and convert them into electrical signals. Unlike the receptors in your skin that respond to external stimuli, proprioceptors monitor internal conditions, the length of your muscles, the tension in your tendons, the angles of your joints. This internal sensing allows your brain to maintain an accurate, real-time representation of your body's configuration in space. The most important proprioceptors are muscle spindles, specialized sensory organs embedded within your skeletal muscles. Each muscle in your body contains dozens to hundreds of these spindles, depending on the muscle's size and the precision of control it requires. Muscles that perform fine movements, like those in your fingers, or the muscles that control eye movement, have high densities of muscle spindles. Muscles involved in cruder movements have fewer spindles. A muscle spindle is a remarkable structure. It consists of several specialized muscle fibers, called intrafusal fibers, wrapped in a connective tissue capsule. The intrafusal fibers are thinner and shorter than the regular muscle fibers, called extrafusal fibers, that generate the force for movement. Wrapped around the central region of each intrafusal fiber is a sensory nerve ending. When the muscle is stretched, the intrafusal fibers stretch as well, deforming the sensory nerve endings and opening mechanically gated ion channels. This generates electrical signals that travel along sensory neurons to the spinal cord and brain. Muscle spindles detect changes in muscle length. When a muscle lengthens, the spindles are stretched and they increase their firing rate. When a muscle shortens, the stretch on the spindles decreases and their firing rate diminishes. This provides your nervous system with continuous information about muscle length and, by extension, the position of your limbs. The sensitivity of muscle spindles is extraordinary. They can detect length changes of less than a millimeter. Muscle spindles also have a motor component. Small motor neurons called gamma motor neurons innervate the intrafusal fibers, causing them to contract. This contraction does not contribute to the force generated by the muscle as a whole, but instead adjusts the sensitivity of the spindle. When gamma motor neurons activate the intrafusal fibers, they take up slack in the spindle, maintaining its sensitivity even as the muscle shortens. This allows muscle spindles to provide useful information throughout the entire range of muscle lengths. The second major type of proprioceptor is the Golgi tendon organ, located at the junction between muscle and tendon. While muscle spindles detect changes in muscle length, Golgi tendon organs detect changes in muscle tension. When a muscle contracts and generates force, or when a muscle is passively stretched with force, the tension is transmitted to the tendon. This tension compresses the Golgi tendon organ, deforming the sensory nerve endings within it and generating action potentials. Golgi tendon organs are exquisitely sensitive to force. They can detect changes in tension produced by the contraction of a single motor unit, one motor neuron, and the small group of muscle fibers it controls. This sensitivity allows your nervous system to monitor not just where your limbs are, but also how much force your muscles are generating. This information is crucial for controlling movement strength and preventing injury from excessive force. The third category of proprioceptors consists of receptors located in and around joints. These include Ruffini endings, Pacinian corpuscles, and free nerve endings embedded in joint capsules and ligaments. Joint receptors respond to mechanical deformation when joints are moved to extreme positions or when ligaments are stretched. While muscle spindles and Golgi tendon organs provide most of the information about limb position during normal movements, joint receptors contribute information about joint angle, particularly at the extremes of the range of motion. Together, muscle spindles, Golgi tendon organs, and joint receptors create a comprehensive sensory system that monitors every aspect of your body's mechanical state. At any given moment, thousands of proprioceptors throughout your body are generating signals. These signals travel along large, heavily myelinated sensory neurons that conduct information rapidly toward your spinal cord. When proprioceptive signals enter the spinal cord through the dorsal roots, They follow the same pathway as tactile information from the skin, the dorsal column medial lemniscus pathway. The axons of proprioceptive neurons ascend in the dorsal columns without crossing, maintaining strict organization with information from the lower body traveling medially and information from the upper body traveling laterally. These axons travel all the way up the spinal cord to the medulla in the brainstem. In the medulla, proprioceptive fibers synapse in the same nuclei that receive tactile input, the gracile nucleus for the lower body and the cuniate nucleus for the upper body. From these nuclei, second-order neurons cross to the opposite side of the brainstem and ascend through the medial lemniscus to the thalamus. The crossing means that proprioceptive information from the left side of your body is processed primarily by the right hemisphere of your brain, and vice versa. The thalamus relays proprioceptive information to the primary somatosensory cortex, where it is integrated with tactile input to create a unified representation of your body. In the somatosensory cortex, neurons respond to specific combinations of joint angles, muscle lengths, and skin contact, building a dynamic model of your body's position and configuration. This model is constantly updated as you move, providing the foundation for motor control and spatial awareness. Proprioceptive information does not only reach the somatosensory cortex, it also travels to the cerebellum, a large structure at the back of your brain beneath the occipital lobe. The cerebellum receives massive amounts of proprioceptive input from muscles, tendons, and joints throughout your body. This information is essential for the cerebellum's primary function, coordinating movement and maintaining balance. The cerebellum compares the proprioceptive feedback it receives with motor commands sent from the cerebral cortex, detecting any discrepancies between intended and actual movements. When a mismatch is detected, the cerebellum generates corrective signals that adjust ongoing movements in real time. Without proprioception, movement becomes slow, uncertain, and inaccurate. People who lose proprioception due to disease or injury must rely entirely on vision to guide their movements. They can move their limbs when they can see them, but if they close their eyes or look away, control deteriorates dramatically. Walking becomes a conscious, deliberate process that requires constant visual monitoring of the ground and the feet. Reaching for objects becomes a series of trial and error adjustments rather than a smooth, automatic action. The loss reveals how much you normally depend on proprioception for even the simplest daily activities. The integration of proprioception with other senses is continuous and essential. Vision and proprioception work together to determine where objects are relative to your body. When you see a cup on a table and reach for it, your brain combines visual information about the cup's location with proprioceptive information about your arm's position to guide your hand to the target. If proprioception is impaired, reaching becomes visually guided throughout, requiring you to watch your hand approach the object rather than looking away once the movement begins. Proprioception also interacts with the vestibular system, the sensory apparatus in your inner ear that detects head movement and orientation. Together, proprioception and vestibular input allow your brain to distinguish between movements of your head relative to your body and movements of your entire body in space. When you turn your head, proprioceptors in your neck detect the rotation, while vestibular receptors in your inner ear detect the change in head orientation. Your brain combines these signals to update your internal representation of where you are looking and how your body is positioned. The sense of effort is closely related to proprioception. When you lift an object, you perceive not only its weight, but also the muscular effort required to hold it. This perception arises partly from proprioceptive feedback, the tension detected by Golgi tendon organs, and the muscle activation sensed by muscle spindles, and partly from corollary discharge, a copy of the motor commands sent by your brain. Your brain compares the motor command with the resulting proprioceptive feedback to determine how much effort the movement required. This is why an object feels lighter when you lift it yourself than when someone else lifts your arm. The presence of your own motor command changes the perception of effort, Proprioception develops early in life and continues to be refined through experience. Infants initially have limited proprioceptive awareness, but as they move, their brains learn to associate patterns of proprioceptive input with specific body configurations. This learning process is crucial for developing motor skills. Athletes, dancers, and musicians develop highly refined proprioception through years of practice, allowing them to perform complex movements with precision and fluidity. Their brains have learned to interpret subtle proprioceptive signals and to integrate them seamlessly with motor control. Proprioceptive sensitivity declines with age. Older adults show reduced accuracy in detecting limb position and muscle tension, which contributes to decreased balance and increased risk of falls. This decline may result from changes in the proprioceptors themselves, reduced numbers of sensory neurons, or changes in the central processing of proprioceptive information. Maintaining physical activity can help preserve proprioceptive function and reduce age-related decline. Right now, as you rest here, proprioceptors throughout your body are active. Muscle spindles are detecting the length of every muscle. Golgi tendon organs are monitoring tension in your tendons. Joint receptors are providing information about limb angles. These signals are traveling up your spinal cord, crossing in your brainstem, relaying through your thalamus, and arriving at your somatosensory cortex and cerebellum. Your brain is constructing a detailed representation of your body's position, where your arms lie, how your legs are bent, the angle of your neck, the position of your fingers. Muscle spindles detect changes in muscle length. When a muscle lengthens, the spindles are stretched, and they increase their firing rate. When a muscle shortens, the stretch on the spindles decreases, and their firing rate diminishes. This provides your nervous system with continuous information about muscle length and, by extension, the position of your limbs. The sensitivity of muscle spindles is extraordinary. They can detect length changes of less than a millimeter. Muscle spindles also have a motor component. Small motor neurons called gamma motor neurons innervate the intrafusal fibers, causing them to contract. This contraction does not contribute to the force generated by the muscle as a whole, but instead adjusts the sensitivity of the spindle. When gamma motor neurons activate the intrafusal fibers, they take up slack in the spindle, maintaining its sensitivity even as the muscle shortens. This allows muscle spindles to provide useful information throughout the entire range of muscle lengths. The second major type of proprioceptor is the Golgi tendon organ, located at the junction between muscle and tendon. While muscle spindles detect changes in muscle length, Golgi tendon organs detect changes in muscle tension. When a muscle contracts and generates force, or when a muscle is passively stretched with force, the tension is transmitted to the tendon. This tension compresses the Golgi tendon organ, deforming the sensory nerve endings within it and generating action potentials. Golgi tendon organs are exquisitely sensitive to force. They can detect changes in tension produced by the contraction of a single motor unit, one motor neuron, and the small group of muscle fibers it controls. This sensitivity allows your nervous system to monitor not just where your limbs are, but also how much force your muscles are generating. This information is crucial for controlling movement strength and preventing injury from excessive force. The third category of proprioceptors consists of receptors located in and around joints. These include Ruffini endings, Pacinian corpuscles, and free nerve endings embedded in joint capsules and ligaments. Joint receptors respond to mechanical deformation when joints are moved to extreme positions or when ligaments are stretched. While muscle spindles and Golgi tendon organs provide most of the information about limb position during normal movements, joint receptors contribute information about joint angle, particularly at the extremes of the range of motion. Together, muscle spindles, Golgi tendon organs, and joint receptors create a comprehensive sensory system that monitors every aspect of your body's mechanical state. At any given moment, thousands of proprioceptors throughout your body are generating signals. These signals travel along large, heavily myelinated sensory neurons that conduct information rapidly toward your spinal cord. When proprioceptive signals enter the spinal cord through the dorsal roots, they follow the same pathway as tactile information from the skin, the dorsal column medial lemniscus pathway. The axons of proprioceptive neurons ascend in the dorsal columns without crossing, maintaining strict organization with information from the lower body traveling medially and information from the upper body traveling laterally. These axons travel all the way up the spinal cord to the medulla in the brainstem. In the medulla, proprioceptive fibers synapse in the same nuclei that receive tactile input, the gracile nucleus for the lower body and the cuneate nucleus for the upper body. From these nuclei, second-order neurons cross to the opposite side of the brainstem and ascend through the medial lemniscus to the thalamus. The crossing means that proprioceptive information from the left side of your body is processed primarily by the right hemisphere of your brain, and vice versa. The thalamus relays proprioceptive information to the primary somatosensory cortex, where it is integrated with tactile input to create a unified representation of your body. In the somatosensory cortex, neurons respond to specific combinations of joint angles, muscle lengths, and skin contact, building a dynamic model of your body's position and configuration. This model is constantly updated as you move, providing the foundation for motor control and spatial awareness. Proprioceptive information does not only reach the somatosensory cortex, it also travels to the cerebellum, a large structure at the back of your brain beneath the occipital lobe. The cerebellum receives massive amounts of proprioceptive input from muscles, tendons, and joints throughout your body. This information is essential for the cerebellum's primary function, coordinating movement and maintaining balance. The cerebellum compares the proprioceptive feedback it receives with motor commands sent from the cerebral cortex, detecting any discrepancies between intended and actual movements. When a mismatch is detected, the cerebellum generates corrective signals that adjust ongoing movements in real time. Without proprioception, movement becomes slow, uncertain, and inaccurate. People who lose proprioception due to disease or injury must rely entirely on vision to guide their movements. They can move their limbs when they can see them, but if they close their eyes or look away, control deteriorates dramatically. Walking becomes a conscious, deliberate process that requires constant visual monitoring of the ground and the feet. Reaching for objects becomes a series of trial and error adjustments rather than a smooth, automatic action. The loss reveals how much you normally depend on proprioception for even the simplest daily activities. The integration of proprioception with other senses is continuous and essential. Vision and proprioception work together to determine where objects are relative to your body. When you see a cup on a table and reach for it, your brain combines visual information about the cup's location with proprioceptive information about your arm's position to guide your hand to the target. If proprioception is impaired, reaching becomes visually guided throughout, requiring you to watch your hand approach the object rather than looking away once the movement begins. Proprioception also interacts with the vestibular system, the sensory apparatus in your inner ear that detects head movement and orientation. Together, proprioception and vestibular input allow your brain to distinguish between movements of your head relative to your body and movements of your entire body in space. When you turn your head, proprioceptors in your neck detect the rotation, while vestibular receptors in your inner ear detect the change in head orientation. Your brain combines these signals to update your internal representation of where you are looking and how your body is positioned. The sense of effort is closely related to proprioception. When you lift an object, you perceive not only its weight, but also the muscular effort required to hold it. This perception arises partly from proprioceptive feedback, the tension detected by Golgi tendon organs, and the muscle activations sensed by muscle spindles, and partly from corollary discharge, a copy of the motor commands sent by your brain. Your brain compares the motor command with the resulting proprioceptive feedback to determine how much effort the movement required. This is why an object feels lighter when you lift it yourself than when someone else lifts your arm. The presence of your own motor command changes the perception of effort. Proprioception develops early in life and continues to be refined through experience. Infants initially have limited proprioceptive awareness, but as they move, their brains learn to associate patterns of proprioceptive input with specific body configurations. This learning process is crucial for developing motor skills. Athletes, dancers, and musicians develop highly refined proprioception through years of practice, allowing them to perform complex movements with precision and fluidity. Their brains have learned to interpret subtle proprioceptive signals and to integrate them seamlessly with motor control. Proprioceptive sensitivity declines with age. Older adults show reduced accuracy in detecting limb position and muscle tension, which contributes to decreased balance and increased risk of falls. This decline may result from changes in the proprioceptors themselves, reduced numbers of sensory neurons, or changes in the central processing of proprioceptive information. Maintaining physical activity can help preserve proprioceptive function and reduce age-related decline. Right now, as you rest here, proprioceptors throughout your body are active. Muscle spindles are detecting the length of every muscle. Golgi tendon organs are monitoring tension in your tendons. Joint receptors are providing information about limb angles. These signals are traveling up your spinal cord, crossing in your brainstem, relaying through your thalamus, and arriving at your somatosensory cortex and cerebellum. Your brain is constructing a detailed representation of your body's position, where your arms lie, how your legs are bent, the angle of your neck, the position of your fingers. The three semicircular canals on each side of your head work together as a set, with each canal most sensitive to rotation in its particular plane. The horizontal canals detect rotation when you turn your head left or right, as if shaking your head no. The anterior and posterior vertical canals detect rotation when you tilt your head forward and backward or tip it side to side. Because the canals are oriented in three perpendicular planes, any rotation of your head, regardless of its direction, will maximally stimulate one canal while having lesser effects on the others. Your brain interprets the pattern of activity across all 6 canals, 3 on each side, to determine the direction and speed of head rotation. The semicircular canals detect angular acceleration, the change in rotational velocity. When you begin turning your head, the canals signal the start of rotation. When you rotate at a constant speed, the endolymph eventually catches up with the canal walls. The cupula returns to its resting position, and the signal diminishes. When you stop rotating, the endolymph continues moving briefly, bending the cupula in the opposite direction and signaling deceleration. This is why you may feel dizzy when you stop spinning after rotating continuously. Your vestibular system is signaling that you are now rotating in the opposite direction, even though you have stopped. The otolith organs, the utricle and the saccule, detect linear acceleration and the orientation of your head relative to gravity. These structures are located in the vestibule, the central chamber of the inner ear between the semicircular canals and the cochlea. Each otolith organ contains a patch of hair cells embedded in a gelatinous layer. Resting on top of this gel are thousands of tiny calcium carbonate crystals called otoliths, or ear stones. These crystals are denser than the surrounding fluid and tissue, giving them significant mass. When your head tilts or when you accelerate in a straight line, gravity and inertia act on the otoliths, causing them to shift position and bend the stereocilia of the hair cells beneath them. The utricle is oriented horizontally when your head is upright, making it sensitive to tilting your head to the side and to horizontal acceleration, such as when you start walking forward or riding in a car. The saccule is oriented vertically, making it sensitive to vertical acceleration, such as when you jump or ride in an elevator. Together, the utricle and saccule provide information about the direction and magnitude of linear acceleration and about the static position of your head relative to gravity. The hair cells in the vestibular system are among the most sensitive mechanoreceptors in your body. They can detect head movements as small as a fraction of a degree and accelerations that would be imperceptible through any other sense. This sensitivity is essential because maintaining balance and stabilizing your gaze require detecting subtle shifts in head position before they become large enough to cause problems. The signals from the vestibular hair cells travel along the vestibular nerve, which joins with the cochlear nerve to form the vestibular-cochlear nerve, the eighth cranial nerve. The vestibular nerve fibers enter the brainstem and synapse in the vestibular nuclei, a group of 4 interconnected nuclei located in the pons and medulla. These nuclei serve as integration centers, receiving input not only from the vestibular organs, but also from the visual system, proprioceptors in the neck and body, and the cerebellum. From the vestibular nuclei, information flows to multiple destinations throughout the nervous system. Some projections travel to the spinal cord, influencing the motor neurons that control posture and balance. These vestibulospinal pathways generate reflexes that automatically adjust your muscle tone and body position to maintain balance when your head moves. If you start to fall to the right, vestibular signals trigger contractions in muscles on the left side of your body to counteract the fall. All without conscious thought, other projections from the vestibular nuclei travel to the nuclei that control eye movements. These connections underlie the vestibulo-ocular reflex, or VOR, one of the fastest and most precise reflexes in your body. The VOR stabilizes your gaze by moving your eyes in the opposite direction to your head movement. When you turn your head to the right, the VOR rotates your eyes to the left by an exactly equal amount, keeping your eyes fixed on a target despite the head movement. This reflex is so effective that you can read text or recognize faces while your head is moving, something that would be impossible if your eyes moved with your head. The VOR operates with a latency of only about 5 to 10 milliseconds, making it one of the fastest reflexes in the human body. This speed is essential because visual processing is relatively slow. By the time your visual system could detect that an object's image is moving across your retina and generate a corrective eye movement, the object would have moved out of your field of sharp vision. The VOR anticipates the need for eye movement based on vestibular signals about head motion, generating the compensatory eye movement almost instantaneously. Vestibular information also projects to the cerebellum, particularly to a region called the vestibular cerebellum. The cerebellum uses vestibular input to coordinate balance and to calibrate the VOR. If your VOR becomes inaccurate, perhaps because you get new glasses that magnify or minify the visual world, your cerebellum can adjust the gain of the reflex over time to restore accurate gaze stabilization. This adaptability allows your vestibular system to remain effective despite changes in your body or sensory apparatus. Finally, vestibular signals reach the cerebral cortex, where they contribute to your conscious sense of spatial orientation and self-motion. The primary cortical areas that process vestibular information are located in the parietal lobe and the junction between the parietal and temporal lobes. These regions integrate vestibular input with visual and proprioceptive information to create your sense of where you are in space and how you are moving. The integration of vestibular, visual and proprioceptive information is essential for balance and spatial orientation. Your brain continuously compares signals from these 3 sensory systems, and when they are consistent, they reinforce each other, creating a stable sense of your body's position and motion. When you stand still on solid ground with your eyes open, vision tells you that you are stationary. Proprioceptors in your legs and trunk confirm that your body is upright, and your vestibular system detects no significant head movement or tilt. All 3 systems agree, and balance is easily maintained. But when sensory information conflicts, balance becomes more difficult. Standing on a moving platform with your eyes closed removes visual information and creates a mismatch between proprioceptive signals from your legs, which detect the platform's movement, and vestibular signals, which may or may not detect motion depending on how the platform moves. Your brain must determine which signals to trust, and this uncertainty can lead to instability. Similarly, when visual input suggests motion, but vestibular input indicates no movement, as when watching a movie, filmed from a moving vehicle, the sensory conflict can produce feelings of disorientation or motion sickness. Motion sickness arises precisely from mismatches between vestibular, visual, and proprioceptive signals. When you read in a car, your vestibular system detects the accelerations and turns, but your eyes, focused on the stationary book, signal no movement. This conflict generates nausea and discomfort, symptoms that likely evolved as a defense against neurotoxins that disrupt sensory processing. Virtual reality can produce similar effects when visual motion is not accompanied by corresponding vestibular stimulation. The relative weighting your brain gives to visual, vestibular, and proprioceptive information can shift depending on context and reliability. When standing on an unstable surface, your brain relies more heavily on vestibular and visual input because proprioceptive signals from your feet are less reliable. When vestibular function is impaired, your brain increases reliance on vision and proprioception to maintain balance. This reweighting process demonstrates the nervous system's flexibility in using available sensory information to achieve stable perception and motor control. Right now, your vestibular system is monitoring your head's position and any small movements you make. Fluid in your semicircular canals tracks rotations. Otoliths in your utricle and saccule respond to gravity and any linear accelerations. Hair cells are generating continuous streams of signals, maintaining a baseline firing rate that increases or decreases with movement. These signals travel to your brainstem, triggering reflexes, stabilizing your gaze, maintaining your posture. Vestibular function is so fundamental to normal experience that you are rarely aware of it unless it malfunctions. When the vestibular system fails, the consequences are immediately apparent and deeply disorienting. Vertigo, the sensation that you or the world around you is spinning, results from abnormal vestibular signals that your brain interprets as rotation, even though no rotation is occurring. This false sense of motion can be so powerful that it makes standing or walking impossible and is often accompanied by nausea and vomiting. One common vestibular disorder is benign paroxysmal positional vertigo, or BPPV, which occurs when otoliths become dislodged and float into one of the semicircular canals. When you move your head, these displaced crystals shift position and stimulate the canal inappropriately, generating signals that falsely indicate rotation. The result is sudden, intense vertigo triggered by specific head positions. BPPV can often be treated with physical maneuvers that reposition the otoliths back into the utricle or saccule. Meniere's disease is another vestibular disorder characterized by episodes of vertigo, hearing loss, and tinnitus. It results from abnormal fluid accumulation in the inner ear, which distorts the vestibular and auditory signals. Vestibular neuritis, inflammation of the vestibular nerve, causes sudden onset of severe vertigo that can last for days, leaving individuals unable to walk or stand without assistance. These conditions reveal how much you depend on normal vestibular function for basic activities. The vestibular system begins developing before birth and continues to mature throughout childhood. Newborn infants already show vestibular reflexes, including the VOR, though these reflexes are not yet fully calibrated. As infants grow and begin to move their heads, sit up, and eventually walk, their vestibular system refines its responses through experience. The integration of vestibular input with vision and proprioception improves gradually, supporting the development of balance and coordinated movement. Like other sensory systems, vestibular function declines with age. The number of hair cells in the semicircular canals and otolith organs decreases, and the remaining hair cells become less sensitive. Vestibular nerve fibers degenerate, reducing the amount of information reaching the brain. These changes contribute to balance problems in older adults, increasing the risk of falls and injuries. Fortunately, the nervous system's ability to reweight sensory information means that enhanced reliance on vision and proprioception can partially compensate for declining vestibular function. Balance training and vestibular rehabilitation can improve stability even when vestibular function is impaired. By challenging the balance system with specific exercises, you can enhance the remaining vestibular function and strengthen the integration of visual and proprioceptive information. These interventions demonstrate the plasticity of the sensory systems involved in balance and spatial orientation. The vestibular system's influence extends beyond balance and spatial orientation. Vestibular signals contribute to autonomic functions affecting heart rate, blood pressure, and respiration. This is why changes in position, such as standing up quickly, require vestibular input to trigger appropriate cardiovascular adjustments. Vestibular input also influences cognitive functions, including spatial memory and navigation. The hippocampus, a brain region critical for memory, receives vestibular information and uses it to create cognitive maps of the environment. Astronauts in microgravity experience profound vestibular disorientation because the otolith organs no longer respond to gravity in the usual way. The semicircular canals still detect head rotation, but the absence of gravitational input from the utricle and saccule creates a sensory void. The brain must adapt to this altered sensory environment, reweighting its reliance on visual and proprioceptive input. Upon returning to Earth, astronauts experience readaptation challenges as their vestibular system recalibrates to the presence of gravity. Your vestibular system, proprioceptive system, visual system, and somatosensory system work together seamlessly to provide your sense of being a physical body occupying space. These senses tell you where you are, how you are oriented, whether you are moving, and how your body is configured. They allow you to navigate through the world, to maintain balance, to coordinate movement, to interact with objects and surfaces. But there is another type of sensation, one that is not about position or motion but about harm, a sensation that warns you when something threatens your body's integrity, that motivates you to withdraw from danger, that captures your attention with urgency and unpleasantness. This is pain, a protective signal that no organism can afford to ignore. From the sense of where you are in space, we turn now to the sense that tells you when something is wrong. Pain is unlike any other sensation you experience. It does not simply inform you about the world. It warns you, motivates you, demands your attention. While other senses can be pleasant or neutral, pain is inherently aversive, designed by evolution to protect your body from harm. It is not merely a sensory signal, but a complex experience that combines physical sensation with emotional distress and urgent behavioral drive. Without pain, you would be in constant danger, unable to detect injury, unaware when tissues are being damaged. The specialized receptors that detect potentially harmful stimuli are called nociceptors. Unlike the encapsulated receptors found in your skin for touch, or the elaborate structures in your eyes and ears, nociceptors are free nerve endings, unspecialized terminals of sensory neurons that extend into tissues throughout your body. They are found in your skin, muscles, joints, bones, and internal organs. These endings contain specialized ion channels, particularly transient receptor potential channels, or TRP channels, that respond to specific types of harmful stimuli. Nociceptors detect three main categories of harmful stimuli: mechanical, thermal, and chemical. Mechanical nociceptors respond to intense pressure or tissue deformation that threatens to cause or has caused damage. Thermal nociceptors activate at extreme temperatures, heat above approximately 45 degrees Celsius, or cold, below about 5 degrees Celsius, temperatures that can damage tissues. Chemical nociceptors respond to substances released by damaged cells or inflammatory processes, including hydrogen ions, potassium, bradykinin, histamine, prostaglandins, and many others. Many nociceptors are polymodal, meaning they can respond to multiple types of harmful stimuli. When tissue is damaged, cells release a complex mixture of inflammatory mediators, sometimes called the inflammatory soup. These substances activate nociceptors directly and also sensitize them, lowering their threshold for activation. This is why injured tissue becomes more sensitive to pain, a phenomenon called peripheral sensitization. After you burn your finger, Even light touch or mild warmth can trigger pain because the nociceptors in that area have become hyperexcitable. This increased sensitivity serves a protective function, encouraging you to protect the injured area while it heals. When a nociceptor is activated, ion channels in the nerve ending open, allowing sodium and calcium ions to flow into the cell. This depolarizes the nerve ending, and if the depolarization is strong enough, it triggers action potentials that propagate along the sensory neuron toward the spinal cord. The signals travel along two main types of nerve fibers, distinguished by their diameter and whether they are myelinated. Delta fibers are relatively large and lightly myelinated, allowing them to conduct signals rapidly at speeds of 5 to 30 meters per second. These fibers carry information about sharp, localized, immediate pain, the kind you feel when you stub your toe or touch something hot. The rapid conduction ensures that you become aware of the injury quickly and can react to remove yourself from danger. This fast pain is often described as sharp, pricking, or stinging. C fibers are smaller and unmyelinated, conducting signals much more slowly at speeds of only 0.5 to 2 meters per second. These fibers carry information about dull, aching, throbbing pain, the kind that follows the initial sharp pain and can persist for extended periods. C fiber pain is often poorly localized and may be accompanied by feelings of nausea or emotional distress. This slow pain serves to remind you that tissue has been damaged and needs protection while it heals. The dual pain system explains a common experience. When you injure yourself, you often feel two distinct waves of pain. The first is sharp and immediate, arriving within a fraction of a second via A delta fibers. The second is duller and builds more slowly, arriving seconds later via C fibers. This temporal separation reflects the different conduction velocities of the two fiber types. The cell bodies of pain neurons, like those of other sensory neurons, are located in the dorsal root ganglia, small clusters of neurons just outside the spinal cord. From the cell body, one axon extends to the periphery, ending in the free nerve endings that serve as nociceptors, while another axon extends into the spinal cord. When action potentials reach the spinal cord, they enter through the dorsal roots and synapse in the dorsal horn, the posterior region of the spinal cord's grey matter. The dorsal horn is organized into layers called laminae, each receiving input from different types of sensory fibers. Delta fibers primarily synapse in laminae I and V, while C fibers synapse in laminae I and T. Here, the first-order pain neurons release neurotransmitters, including glutamate and substance P, onto second-order neurons. These second-order neurons can be nociceptive specific, responding only to painful stimuli, or wide dynamic range neurons, responding to both painful and non-painful stimuli, but firing more intensely for pain. The dorsal horn is not simply a relay station, but a site of significant processing and modulation. Interneurons within the dorsal horn can amplify or suppress pain signals before they are transmitted to the brain. Excitatory interneurons enhance pain transmission, while inhibitory interneurons reduce it. This modulation creates a gate that can open to allow pain signals through or close to block them. The balance between excitation and inhibition in the dorsal horn determines how much pain information reaches your brain. From the dorsal horn, second-order pain neurons send their axons across the midline of the spinal cord to the opposite side. This crossing occurs at the same spinal level where the neurons enter, meaning that pain information from the left side of your body is processed by the right side of your brain almost immediately. After crossing, the axons ascend through the white matter of the spinal cord in a pathway called the spinothalamic tract. The spinothalamic tract actually consists of two components with different functions. The lateral spinothalamic tract, sometimes called the neospinothalamic pathway, carries information about the sensory discriminative aspects of pain, its location, intensity, and quality. This pathway projects to the ventral posterior nucleus of the thalamus and then to the primary and secondary somatosensory cortices. These cortical areas allow you to determine where the pain is coming from and how intense it is. The medial spinothalamic tract, also called the paleospinothalamic pathway, carries information about the effective motivational aspects of pain, its unpleasantness, and emotional impact. This pathway is more diffuse, projecting to multiple regions including the medial thalamus, the brainstem reticular formation, and the periaqueductal gray. From the medial thalamus, signals project to the anterior cingulate cortex, the insula, and the prefrontal cortex. These areas generate the emotional and autonomic responses to pain, the distress, the anxiety, the increased heart rate, and blood pressure. This dual pathway system explains why pain has both sensory and emotional dimensions. You can perceive where pain is located and how intense it is through the lateral pathway, while simultaneously experiencing the unpleasantness and urgency of pain through the medial pathway. The two components are separable. Certain brain lesions or surgical interventions can reduce the emotional distress of pain while leaving the sensory perception intact. The thalamus serves as a critical relay point for pain information, just as it does for other sensory modalities. But unlike touch or vision, where thalamic processing is relatively straightforward, pain signals are distributed to multiple thalamic nuclei, each projecting to different cortical and subcortical regions. This widespread distribution reflects the multidimensional nature of pain as both a sensation and an experience. When pain signals reach the cerebral cortex, they activate a network of brain regions collectively called the pain matrix. This network includes the primary and secondary somatosensory cortices, the insula, the anterior cingulate cortex, and the prefrontal cortex. Each region contributes different aspects to the pain experience. The somatosensory cortices process the location and intensity. The insula contributes to the subjective feeling of pain and integrates it with interoceptive awareness of your body's internal state. The anterior cingulate cortex is involved in the emotional and attentional aspects of pain, generating the sense of unpleasantness and suffering. The prefrontal cortex plays a particularly interesting role. This region, responsible for higher cognitive functions, modulates pain perception based on context, expectation, and attention. The prefrontal cortex can enhance or suppress pain intensity depending on what you believe about the pain, what it means, and whether you are focused on it. This is the neural basis for phenomena like pain catastrophizing, where anxiety and negative expectations intensify pain experience, and for the placebo effect, where belief in a treatment's effectiveness can reduce pain even when the treatment has no direct analgesic properties. Pain is also strongly connected to memory and emotion through the limbic system. The amygdala, a structure involved in fear and emotional learning, receives pain signals and associates them with the context in which they occurred. This creates emotional memories of pain, which can influence future pain experiences. If you were injured in a particular place or situation, returning to that context may increase your pain sensitivity or anxiety about pain. The subjective intensity of pain does not correlate perfectly with the intensity of the stimulus that caused it. Two people can experience the same injury but report very different levels of pain. One person may find a stimulus excruciating while another finds it merely uncomfortable. These differences arise from variations in pain pathways, genetic factors affecting nociceptor sensitivity and pain processing, past experiences with pain, psychological state, and the context in which pain occurs. Your body also has built-in systems for reducing pain. Descending pathways from the brainstem can suppress pain signals in the dorsal horn of the spinal cord. The periaqueductal gray in the midbrain and the rostral ventromedial medulla in the brainstem send signals down the spinal cord that activate inhibitory interneurons, closing the pain gate and reducing transmission of pain signals to the brain. These descending inhibitory pathways are activated by stress, intense focus, and the body's own opioid-like chemicals called endorphins. This endogenous pain modulation system explains phenomena like stress-induced analgesia, where soldiers injured in battle or athletes injured during competition may not feel pain until the stressful situation has passed. The brain suppresses pain signals when immediate action is more important than attending to injury. Once safety is restored, the descending inhibition diminishes and pain returns to conscious awareness. Chronic pain represents a breakdown in the normal protective function of pain. While acute pain serves as a warning signal that motivates protective behavior and healing, chronic pain persists long after tissue has healed or occurs without any clear injury. Chronic pain involves changes at multiple levels of the nervous system. Peripheral sensitization continues even after healing, keeping nociceptors hyperexcitable. Central sensitization occurs in the spinal cord, where repeated pain signals cause long-lasting increases in the excitability of dorsal horn neurons. These neurons begin responding to normally innocuous stimuli as though they were painful, a phenomenon called allodynia. The brain itself changes in chronic pain conditions. Brain imaging studies show that people with chronic pain have altered activity in the pain matrix, with some regions becoming hyperactive, while others show reduced function. The descending inhibitory pathways may become less effective, removing a natural brake on pain transmission. The prefrontal cortex may lose its ability to modulate pain, leaving individuals unable to suppress pain signals through attention or cognitive strategies. Chronic pain also has profound effects on emotion, cognition, and quality of life. The constant activation of the effective pain pathways leads to depression, anxiety, and emotional distress. Sleep is disrupted, which further lowers pain threshold and impairs the body's natural pain modulation systems. Attention and memory can be impaired because the brain's resources are constantly diverted to processing pain. The experience of chronic pain becomes self-reinforcing, with emotional distress amplifying pain, which causes more distress, creating a cycle that is difficult to break. Yet pain is not inevitable in every instance of tissue damage. Context, meaning, and attention powerfully shape pain experience. The same injury can produce different amounts of pain depending on whether you understand what caused it, whether you believe it is serious, whether you are distracted by other concerns, and what emotional state you are in. This variability is not weakness or imagination, but reflects the genuine complexity of pain processing, where sensory signals are filtered through multiple levels of modulation before reaching consciousness. Right now, nociceptors throughout your body are monitoring for threats. Most are silent, not firing because no harmful stimuli are present, but some may be generating low levels of activity in response to minor discomforts. Pressure from lying in one position, slight inflammation in a joint, the faint ache of tired muscles. These signals travel to your spinal cord, where they are modulated by local circuits and descending inhibition. Most never reach your conscious awareness because your brain has determined they are not urgent enough to require attention. Pain is a guardian, constantly watching for danger, ready to alert you when protection is needed. It is unpleasant by necessity, designed to capture your attention and motivate action. Without pain, you would be vulnerable, unable to sense injury, unaware of damage until it became catastrophic. With it, you have an early warning system, a sensation that, however unwelcome, serves the fundamental purpose of keeping you alive. But your senses do not work in isolation. The visual, auditory, tactile, chemical, proprioceptive, vestibular, and pain signals all converge, all interact, all contribute to a unified experience of reality. Your brain does not process each sense separately and then assemble them like pieces of a puzzle. Instead, sensory information flows together, influences each other, creates an integrated whole greater than the sum of its parts. From the individual pathways of sensation, we turn now to how they merge, how they interact, how your brain binds them into a seamless perceptual world. Your brain does not experience sight in one location, sound in another, and touch in yet another, keeping each sense neatly separated. Instead, sensory information converges, interacts and influences itself across modalities in ways both subtle and profound. What you see changes what you hear, what you touch influences what you see. Your perception of the world is not a collection of independent sensory streams, but a unified, integrated experience built from the continuous interaction of all your senses working together. This multisensory integration happens automatically, outside conscious awareness. Your brain is constantly comparing and combining information from different sensory systems, checking for consistency, resolving conflicts, and using input from one sense to enhance or clarify input from another. The result is a perceptual experience that is more accurate, more reliable, and more informative than any single sense could provide alone. The integration begins at surprisingly early stages of sensory processing. While each sensory system has dedicated pathways and specialized cortical areas, these regions are not isolated. They exchange information through direct connections and through shared projections to higher-order association areas. Even primary sensory cortices, once thought to process only their own modality, receive input from other senses. The primary visual cortex receives tactile and auditory input. The primary auditory cortex is influenced by visual information. This cross-modal connectivity allows each sense to be shaped by others from the earliest stages of cortical processing. One of the most striking demonstrations of multisensory integration is the McGurk effect. In this illusion, you watch a video of someone speaking while the audio is mismatched with the visual lip movements. If the video shows lips forming the syllable ga while the audio plays ba, you often perceive a third syllable entirely, de, that represents a fusion of the visual and auditory information. Your brain cannot help but integrate what it sees with what it hears, even when the two sources conflict. The McGurk effect reveals that speech perception is inherently multisensory, combining auditory input from the voice with visual input from lip movements to create a unified perception of what is being said. This integration of vision and hearing extends beyond speech. When you watch a musician play an instrument, the visual information about when the musician's hand strikes a key or plucks a string influences your perception of when the sound occurs. If the visual and auditory information are slightly misaligned in time, your brain often adjusts one to match the other, creating the perception of synchrony even when the physical stimuli are asynchronous. This temporal binding happens within a window of roughly 100 to 200 milliseconds. If visual and auditory events occur within this window, your brain treats them as arising from the same source and binds them together. The brain uses several principles to determine whether sensory inputs should be integrated. Spatial coincidence is one. Events that occur in the same location are more likely to arise from the same source and are therefore more likely to be bound together. Temporal coincidence is another. Events that happen at the same time are treated as related. The principle of inverse effectiveness also applies. Multisensory integration is strongest when individual sensory signals are weak or ambiguous. When vision is clear and hearing is clear, each sense can operate independently. But when vision is degraded, perhaps due to dim lighting or distance, and hearing is noisy, the combination of the two senses produces a much larger benefit, allowing you to perceive things you could not detect through either sense alone. Touch and vision interact extensively. When you reach for an object you can see, your brain integrates visual information about the object's location with proprioceptive and tactile information from your hand. This integration guides your movements with remarkable precision, but the interaction goes deeper. Visual input can alter tactile perception, making touch feel different depending on what you see. The rubber hand illusion demonstrates this powerfully. When you watch a rubber hand being stroked in synchrony with your real hand, which is hidden from view, you begin to feel as though the touch is occurring on the rubber hand. Your sense of touch has been captured by vision, pulled toward the visible hand even though the actual tactile stimulation is occurring elsewhere. Vision also influences your sense of balance. Your vestibular system provides information about head movement and orientation, but this information is integrated with visual input to create your sense of stability. When you stand still in a room where the walls are slowly moving, you may feel as though you are swaying or falling, even though your vestibular system correctly signals that you are stationary. Vision is so powerful that it can override vestibular input, creating an illusion of self-motion called vection. This is why watching a movie filmed from a moving vehicle can make you feel as though you are moving, and why virtual reality can produce motion sickness when visual motion is not accompanied by corresponding vestibular stimulation. Sound and touch interact as well, particularly for detecting vibrations. When you feel a vibration and hear a corresponding sound simultaneously, your perception of both is enhanced. The brain regions that process touch and hearing are anatomically close and share connections, facilitating this interaction. This is why music can feel more immersive when bass frequencies are strong enough to produce tactile sensations, or why haptic feedback enhances the realism of virtual environments. The convergence of sensory information occurs in multiple brain regions. Association cortices in the parietal, temporal, and frontal lobes receive input from multiple sensory modalities and contain neurons that respond to combinations of sensory stimuli. The superior temporal sulcus, for example, is a multisensory area where visual and auditory information converge. Neurons in this region respond to both visual motion and sound, and some respond preferentially when visual and auditory stimuli are spatially aligned. The posterior parietal cortex integrates visual, tactile, and proprioceptive information to support visually guided reaching and grasping. The integration of sensory information is not static, but adapts based on experience and context. Your brain learns which combinations of sensory signals reliably occur together and strengthens the connections between them. This is why a particular smell can instantly evoke a visual memory of a place, or why hearing a familiar voice can bring to mind the person's face. The associations between senses are built through repeated exposure and become automatic, triggering one another without conscious effort. Attention plays a crucial role in multisensory integration. When you focus attention on one sensory modality, processing in that modality is enhanced while processing in other modalities may be suppressed. But attention can also be captured across modalities. A sudden sound draws your visual attention to the location of the sound. A flash of light in your peripheral vision causes you to turn your head and listen for associated sounds. This cross-modal attentional capture ensures that unexpected events are detected quickly, regardless of which sense first encounters them. The brain's multisensory integration supports rapid and accurate decision-making. When you need to respond to an event, decision signals from different senses unfold in parallel, but converge in motor areas that control action. If both visual and auditory information indicate that a car is approaching, the combined signal reaches the motor system faster and more reliably than either signal alone. This multisensory enhancement of motor responses is one reason why performance is better when multiple senses provide consistent information about the environment. Language and communication rely fundamentally on multisensory integration. When you converse with someone, you are not simply hearing words. You are watching facial expressions, seeing gestures, noting body language, and integrating all of this visual information with the auditory signal. This integration is so automatic that it feels effortless, but it involves extensive coordination between visual and auditory cortices, as well as higher-order language areas in the frontal and temporal lobes. People who communicate using sign language show similar multisensory integration, but with different weightings. Visual information becomes primary, while auditory information may be less relevant. Flavor perception exemplifies multisensory integration at its most complex. What you experience as the taste of food is actually a synthesis of true taste from your tongue, smell from retronasal olfaction, texture from tactile receptors in your mouth, temperature from thermoreceptors, and even pain from receptors that respond to spicy foods. These diverse sensory inputs converge in the orbitofrontal cortex, where neurons respond to specific combinations of taste, smell, and texture. The color and appearance of food also influence flavor perception. A drink that looks red is expected to taste like cherry, and violating this expectation can make the flavor seem wrong, even if the actual taste and smell are pleasant. The timing of multisensory integration is critical. Different senses process information at different speeds, and different types of stimuli within the same sense can have different processing times. Visual processing of motion is faster than visual processing of color. Auditory processing of sudden sounds is faster than visual processing of the same event. Despite these differences, your perception feels synchronized. Your brain compensates for the varying processing speeds, adjusting the timing of signals so that events that occurred simultaneously in the world are perceived as simultaneous. This temporal recalibration can adapt to systematic delays. If you wear earphones that introduce a delay between visual and auditory signals, your brain will gradually adjust, learning to interpret the delayed audio as synchronous with the visual input. After adaptation, removing the delay makes the two senses seem out of sync in the opposite direction, revealing that your brain had recalibrated its expectations. This flexibility allows multisensory integration to remain accurate, despite changes in the environment, or in sensory processing itself. Multisensory integration is not merely additive, but can produce synergistic effects. The response to combined visual and auditory stimuli is often greater than the sum of responses to each stimulus alone. This superadditivity occurs particularly when individual stimuli are weak or ambiguous, as predicted by the principle of inverse effectiveness. In noisy or degraded sensory conditions, multisensory integration becomes especially valuable, allowing you to detect and identify stimuli that would be undetectable through any single sense. The development of multisensory integration begins in infancy and continues into childhood. Newborns show some multisensory responses, but the precision and sophistication of integration improves with experience. Young children gradually learn to use spatial and temporal coincidence to determine which sensory signals belong together. They learn the typical associations between sights and sounds, between textures and visual appearances, between smells and tastes. This learning shapes the multisensory circuitry of the brain, strengthening connections between sensory areas and tuning neurons to respond to specific multisensory combinations. When multisensory integration fails or is disrupted, perception can become fragmented and confusing. Some neurological conditions affect the ability to bind sensory information across modalities. People may hear speech but have difficulty integrating it with lip movements, reducing comprehension in noisy environments. Others may struggle to coordinate visual and proprioceptive information, making visually guided movements clumsy and inaccurate. These deficits reveal how much normal perception depends on the seamless integration of multiple senses. Right now, your brain is integrating sensory information from every active sensory system. Visual input about the dim light in the room combines with tactile input about surfaces touching your body. Auditory input about ambient sounds merges with vestibular information about your head's position. Proprioceptive signals about limb configuration integrate with tactile feedback from pressure points. All of these streams of information are flowing together, influencing each other, creating a unified sense of your body in space. The neural mechanisms underlying multisensory integration involve complex patterns of connectivity and communication between brain regions. Sensory cortices send projections to multisensory convergence zones, where neurons receive input from multiple modalities. These multisensory neurons can perform computations that single sensory neurons cannot, such as detecting spatial correspondence between visual and auditory events or temporal relationships between touch and vision. The output of multisensory areas projects to motor systems, decision-making circuits, and higher cognitive regions, allowing integrated sensory information to guide behavior and thought. The superior colliculus, a structure in the midbrain, exemplifies multisensory integration at a subcortical level. This structure receives visual, auditory, and somatosensory input, and contains neurons that respond to stimuli from multiple modalities. When visual and auditory stimuli occur at the same location, neurons in the superior colliculus respond more strongly than they do to either stimulus alone. These multisensory neurons play a crucial role in orienting responses, turning your head and eyes toward unexpected events. The enhanced response to multisensory stimuli ensures that you detect and orient toward important events quickly and reliably. Multisensory integration also occurs in memory and imagination. When you remember an experience, you often recall not just what you saw, but also what you heard, felt, smelled, and tasted. These sensory components are bound together in memory, associated with each other and with the context in which they occurred. Recalling one sensory aspect of a memory can trigger retrieval of others, allowing you to reconstruct the full, multisensory experience. Similarly, when you imagine a future event, you construct a multisensory simulation, combining visual imagery with imagined sounds, anticipated tactile sensations, and even predicted smells. The phenomenon of synesthesia represents an unusual form of multisensory interaction, where stimulation of one sense automatically triggers experiences in another. Some individuals experience colors when they hear sounds, or taste specific flavors when they see certain shapes. Synesthesia likely arises from atypical connections between sensory areas in the brain, allowing activation in one sensory cortex to spread to others. While synesthesia is relatively rare, it highlights the extensive connectivity between sensory systems and the potential for cross-modal influences. Multisensory integration extends beyond perception to influence emotion and motivation. The emotional impact of an experience is enhanced when multiple senses contribute. A movie is more emotionally engaging with both visuals and soundtrack than with visuals alone. A meal is more satisfying when it appeals to multiple senses: appearance, aroma, texture, taste, and even sound. The limbic system, which processes emotion, receives input from multiple sensory modalities and integrates them to determine the emotional significance of events. The plasticity of multisensory integration means that it can be altered by training or by sensory deprivation. People who are blind show enhanced integration of auditory and tactile information, with auditory signals producing stronger activation in brain areas normally involved in vision. Similarly, people who are deaf show increased reliance on visual information and enhanced visual processing. These changes demonstrate the brain's ability to reorganize sensory processing based on which inputs are available, optimizing integration for the sensory information that remains. Technology increasingly exploits multisensory integration to create immersive experiences. Virtual reality combines visual and auditory information, and increasingly adds tactile feedback to create the sensation of presence in a simulated environment. The effectiveness of these simulations depends on maintaining consistency across sensory modalities and respecting the brain's principles for multisensory binding. When sensory cues are misaligned, when visual motion is not accompanied by vestibular stimulation, for example, the illusion breaks down and discomfort results. Your perception of the world is constructed moment by moment through the integration of sensory information. Each sense contributes its own perspective, its own type of information, its own strengths and limitations. Vision provides high-resolution spatial information, but requires light. Hearing provides temporal information and works in darkness. Touch provides direct contact information. Smell and taste reveal chemical composition. Proprioception and the vestibular system provide information about your body's configuration and motion. Together, these senses create a representation of reality that is richer, more accurate, and more reliable than any single sense could achieve. The seamless integration of your senses creates the unified experience you call perception. But this integrated information must go somewhere, must be processed and interpreted to become conscious experience. The raw sensory data converging in your brain must be transformed into meaning, into recognition, into understanding. From the convergence of senses, we turn now to the places where these signals are interpreted, where sensation becomes perception, where neural activity becomes conscious awareness. The sensory signals traveling through your nervous system are not simply passed along unchanged until they reach some central viewing area in your brain. Instead, at every stage of their journey, these signals are transformed, filtered, and interpreted. By the time sensory information reaches the cerebral cortex, it has already been processed multiple times in the receptors themselves, in the spinal cord or brainstem, in the thalamus. But it is in the cortex, the folded outer layer of your brain, where the most sophisticated analysis occurs. Here, in specialized regions dedicated to different senses, raw sensory data becomes meaningful perception. The cerebral cortex is divided into 4 main lobes on each side of your brain: the frontal, parietal, temporal, and occipital lobes. Each lobe contains regions dedicated to specific functions, and for sensory processing, certain areas have become highly specialized. The occipital lobe, at the back of your brain, houses the visual cortex. The temporal lobe, on the sides, contains the auditory cortex and parts of the olfactory cortex. The parietal lobe, near the top and back, holds the somatosensory cortex, which processes touch, proprioception, and pain. These primary sensory cortices are the first cortical destinations for sensory information, the places where conscious perception begins to emerge. Before sensory signals reach the cortex, most pass through the thalamus, a structure deep in the center of your brain. The thalamus is sometimes called the brain's relay station, but this description understates its importance. The thalamus does not merely forward sensory information but actively processes and modulates it. Different regions of the thalamus receive input from different sensory systems. The lateral geniculate nucleus receives visual information from the retina. The medial geniculate nucleus receives auditory information from the brainstem. The ventral posterior nucleus receives somatosensory information about touch, proprioception, and pain. Only olfaction bypasses the thalamus, projecting directly from the olfactory bulb to the piriform cortex. The thalamus does more than sort and relay sensory signals. It filters them based on their importance and your current state of attention. When you are focused on a task, the thalamus enhances signals relevant to that task while suppressing irrelevant sensory input. When you sleep, the thalamus reduces the flow of sensory information to the cortex, allowing you to remain unconscious despite ongoing sensory stimulation. This gating function is essential for preventing sensory overload and for directing your attention to what matters most in any given moment. From the thalamus, sensory information projects to the primary sensory cortices through organized pathways called thalamocortical radiations. These projections maintain the topographic organization established earlier in the sensory pathways. Visual information arrives at the primary visual cortex with a map of the visual field preserved. Auditory information arrives at the primary auditory cortex with frequency organization maintained. Somatosensory information arrives at the primary somatosensory cortex with a map of the body surface intact. This systematic organization allows the cortex to know not just what type of sensory information is arriving, but where it came from in sensory space. The primary visual cortex, or V1, occupies the posterior pole of the occipital lobe, wrapping around to the medial surface of the brain. This region is also called the striate cortex because of a prominent stripe of white matter running through it. V1 contains a detailed map of the visual field, with each small region of cortex corresponding to a specific location in space. The central region of your vision, the fovea, occupies a disproportionately large area of V1 compared to peripheral vision. This magnification reflects the high density of photoreceptors in the fovea and the importance of central vision for detailed perception. Neurons in V1 are organized into columns perpendicular to the cortical surface. Cells within a single column respond to stimuli from the same small region of visual space and often prefer the same orientation of edges or lines. Adjacent columns respond to adjacent regions of space or to slightly different orientations. This columnar organization creates a systematic map, not only of visual space, but also of visual features like orientation, spatial frequency, and ocular dominance. The discovery of this organization earned David Hubel and Torsten Wiesel the Nobel Prize in 1981. The primary auditory cortex, or A1, is located on the superior temporal gyrus, the upper ridge of the temporal lobe. Much of A1 is hidden within the lateral fissure, the deep fold that separates the temporal lobe from the frontal and parietal lobes. Like the visual cortex, A1 is organized tonotopically, with neurons responding to low frequencies located in one region and neurons responding to high frequencies in another. This frequency map reflects the organization established in the cochlea and preserved through every relay station along the auditory pathway. A1 neurons do more than simply detect sound frequency. They respond to complex acoustic features including temporal patterns, amplitude modulations, and combinations of frequencies. Some neurons fire selectively for sounds that sweep upward or downward in frequency. Others respond to specific temporal patterns of sound onset and offset. Through this hierarchical processing, A1 begins to extract the features that will be used by higher auditory areas to recognize speech, music, and environmental sounds. The primary somatosensory cortex, or S1, occupies the postcentral gyrus, a ridge of cortex that runs from the top of the brain down the lateral surface. S1 is organized somatotopically, with different body parts represented in different cortical locations. The organization creates the sensory homunculus, a distorted map of the body draped across the cortical surface. Your feet are represented at the top near the midline. Moving down the cortex, you encounter representations of the legs, trunk, arms, hands, face, and lips. The size of each body part's representation corresponds to its sensory importance rather than its physical size. Your hands and lips occupy enormous areas of cortex. Your back and legs occupy relatively small areas. S1 is subdivided into distinct regions called Brodmann areas, each processing different aspects of somatosensory information. Area 3A receives input primarily from muscle and joint receptors, processing proprioception. Area 3B receives input from cutaneous receptors, processing touch. Area 1 processes texture information. Area 2 integrates information about size and shape, combining tactile and proprioceptive input. These subdivisions work in parallel, each extracting different features from the somatosensory input, then sharing information to create a complete representation of what you are touching and how your body is positioned. Beyond the primary sensory cortices lie secondary and association areas where sensory processing becomes increasingly complex and abstract. Information flows forward from the primary areas into regions that extract higher-level features, recognize patterns, and ultimately create meaning from sensory input. In vision, this hierarchical processing extends from V1 through a series of visual areas designated V2, V3, V4, V5, and beyond. Each area specializes in particular aspects of visual analysis. Area V2, which surrounds V1, continues the analysis of form, color, and motion begun in the primary visual cortex. V2 neurons respond to more complex features than V1 neurons, detecting combinations of edges, orientations, and colors that correspond to shapes and contours. From V2, visual information splits into two major processing streams that follow different pathways through the brain. The ventral stream, sometimes called the what pathway, extends from V1 through V2 and V4 into the inferior temporal cortex. This pathway specializes in object recognition and identification. Neurons along the ventral stream respond to progressively more complex visual features. Cells in V4 respond strongly to color and to moderately complex shapes. Neurons in the inferior temporal cortex respond to highly specific objects, including faces, hands, and bodies. Some neurons in this region fire selectively when you see a particular person's face, regardless of viewing angle lighting, or expression. These cells represent the culmination of hierarchical processing that began with simple edge detectors in V1 and ended with neurons that encode the identity of specific individuals. The dorsal stream, called the where or how pathway, extends from V1 through V2 into the parietal cortex. This pathway specializes in spatial processing and visually guided action. Area V5, also known as MT, is a key node in the dorsal stream, containing neurons highly sensitive to motion direction and speed. Damage to V5 produces a rare condition called achinotopsia, in which objects appear frozen in disconnected snapshots rather than moving smoothly. The posterior parietal cortex integrates visual information with proprioceptive and vestibular input to support reaching, grasping, and navigation. Neurons in this region encode the locations of objects relative to your body and the trajectories required to reach them. In the auditory system, similar hierarchical processing occurs beyond A1. Secondary auditory areas surrounding A1 analyze increasingly complex acoustic patterns. Some regions specialize in processing speech sounds, particularly in the left hemisphere. The superior temporal sulcus contains neurons that respond to voice quality and to the emotional tone of speech. Higher auditory areas in the temporal lobe extract semantic meaning from sound, linking acoustic patterns to stored knowledge about words, music, and environmental sounds. The somatosensory system also has secondary cortical areas beyond S1. The secondary somatosensory cortex, S2, lies in a region where the parietal lobe curves down toward the temporal lobe. S2 receives input from S1 and processes more complex tactile information. Unlike S1, where neurons respond primarily to stimulation on one side of the body, many S2 neurons respond to touch on either side, allowing for bilateral integration. This integration is important for tasks requiring coordination of both hands, such as tying knots or manipulating objects. Association cortices are regions that receive input from multiple sensory modalities and integrate them to create unified perceptual experiences. The posterior parietal cortex is a major association area, receiving visual, somatosensory, and auditory input. Neurons here represent space in multiple coordinate systems, combining information about eye position, head position, and body position to create a flexible representation of where objects are in the world. The posterior parietal cortex is essential for spatial attention, allowing you to direct your focus to specific locations and track objects as they move. The temporal parietal junction, where the temporal and parietal lobes meet, is another multisensory association area. This region integrates information about your body's position, the locations of objects around you, and social information about other people. Damage to this area can produce striking deficits in spatial awareness, including neglect syndromes where individuals fail to attend to one side of space despite intact sensory systems. The insula, a region of cortex folded deep within the lateral fissure, integrates multiple sensory and interoceptive signals. The insula receives input from taste, touch, pain, and visceral sensory systems. It plays a crucial role in creating your sense of your body's internal state: hunger, thirst, temperature, pain, emotion. The anterior insula is particularly involved in conscious awareness of bodily sensations and in the subjective feeling of emotions. The orbitofrontal cortex, located on the underside of the frontal lobe just above the eye sockets, is where taste and smell converge with visual and somatosensory information to create flavor perception. Neurons in this region respond to combinations of sensory features that define specific foods. The orbitofrontal cortex also integrates sensory information with reward and motivation, determining which sensory experiences are pleasant or unpleasant, desirable or aversive. Throughout the sensory cortices, neural responses are not fixed, but are modulated by attention, expectation, and context. When you pay attention to a particular stimulus, activity in the corresponding sensory cortex is enhanced. When you expect to see or hear something, the relevant sensory areas become active even before the stimulus appears. This top-down modulation allows your brain to prioritize processing of relevant sensory information and to interpret ambiguous stimuli based on prior knowledge and expectations. The sensory cortices are constantly active, even in the absence of external stimulation. Spontaneous activity in these regions generates the mental imagery you experience when you close your eyes and visualize a scene, when you imagine a sound, or when you recall a tactile sensation. This internal activity draws on the same neural machinery used for processing actual sensory input, activating patterns similar to those produced by real perception. The communication between sensory areas and higher cognitive regions is bidirectional. While sensory information flows forward from primary to secondary to association cortices, there are also massive feedback connections flowing in the opposite direction. These feedback projections from higher areas back to lower areas are actually more numerous than the forward connections. Higher cortical areas send predictions and expectations back to sensory areas, shaping how incoming sensory information is processed. This predictive processing allows your brain to interpret sensory input based on context and prior knowledge, rather than treating each stimulus as entirely new. The prefrontal cortex, the most anterior region of the frontal lobe, plays a crucial role in controlling sensory processing. This region is involved in attention, working memory, and executive control. When you decide to focus on a particular sensory feature, to listen for a specific sound, or to look for a particular object, your prefrontal cortex sends signals to the relevant sensory areas, enhancing their responsiveness. This attentional enhancement can be so strong that it changes which stimuli you consciously perceive. Two people experiencing the same sensory input may perceive different things depending on where they direct their attention. Sensory processing in the cortex is highly plastic, capable of reorganizing based on experience. If you lose sensation in one finger, the region of somatosensory cortex that formerly responded to that finger will gradually begin responding to adjacent fingers instead. This reorganization demonstrates that cortical maps are not fixed, but are continuously shaped by the patterns of input they receive. Musicians who practice extensively show enlarged representations of the fingers they use most. People who read Braille develop enhanced cortical representations of the fingertips used for reading. The visual cortex can also be recruited for other senses when vision is absent. In people who are blind from an early age, the visual cortex responds to tactile and auditory input. This cross-modal plasticity allows the unused visual cortex to support enhanced processing in the remaining senses. Blind individuals who read Braille show activation of the visual cortex while reading, as if the cortex has been repurposed for tactile processing. This remarkable flexibility demonstrates that cortical areas are defined more by their connectivity and function than by any inherent sensory modality. The speed of sensory processing varies across different cortical areas and different types of stimuli. The first neural responses in primary sensory cortices occur within tens of milliseconds of stimulus onset. A visual stimulus activates V1 in about 40 to 60 milliseconds. An auditory stimulus activates A1 in about 10 to 20 milliseconds. But conscious perception takes longer, requiring at least 100 to 200 milliseconds of sustained activity. The initial cortical responses are not sufficient for awareness. They must be amplified and sustained through feedback connections and recurrent processing before they reach consciousness. Different stages of processing extract features at different levels of complexity. Early visual areas encode simple features like edges and colors. Middle-level areas combine these features into shapes and surfaces. High-level areas recognize complete objects and scenes. This hierarchical organization means that damage at different levels produces different deficits. Damage to V1 produces blindness in part of the visual field. Damage to higher visual areas can leave basic vision intact, while impairing the ability to recognize faces, objects, or spatial relationships. The cortical processing of pain involves multiple areas distributed across the brain. The primary and secondary somatosensory cortices encode the location and intensity of pain. The anterior cingulate cortex processes the emotional and attentional aspects of pain, generating the sense of suffering. The insula contributes to the subjective quality of pain and integrates it with other bodily sensations. The prefrontal cortex modulates pain perception based on cognitive factors like expectation and belief. This distributed processing explains why pain is such a multidimensional experience, combining sensory, emotional, and cognitive components. Conscious perception emerges from coordinated activity across multiple cortical areas. No single region is solely responsible for consciousness. Instead, awareness arises from widespread networks that include sensory cortices, association areas, and frontal regions. When you consciously perceive something, activity becomes synchronized across these distributed networks, creating what is sometimes called a neural coalition or global workspace. This synchronized activity allows information to be broadly shared across the brain, making it available for verbal report, memory formation, and decision-making. The transition from unconscious to conscious processing involves changes in both the amount and pattern of cortical activity. Unconscious stimuli produce brief, localized activation in sensory cortices. Conscious stimuli produce sustained, widespread activation that reverberates between cortical areas through feedback connections. This amplification and broadcasting of sensory information is what brings it into awareness. Factors that promote this amplification, such as attention, stimulus strength, and the absence of competing stimuli, make perception more likely to become conscious. Right now, your sensory cortices are processing streams of information. Visual cortex is analyzing the patterns of light entering your eyes. Auditory cortex is processing ambient sounds. Somatosensory cortex is interpreting pressure and temperature from your skin, and proprioceptive signals from your muscles and joints. These processing centers are extracting features, recognizing patterns, integrating information across modalities. They are communicating with each other and with higher cognitive areas, creating the rich, unified perceptual experience that feels so immediate and effortless. But sensory processing does not end with immediate perception. The information flowing through your sensory cortices can be stored, can become memory, can be retrieved later to influence future perception and behavior. From the immediate processing of sensation, we turn now to how sensory experiences become memories, how perception extends across time. Every sensory experience you have ever had leaves a trace: the face of someone you love, the sound of their voice, the scent of rain on summer pavement, the taste of your grandmother's cooking. These are not simply moments that occurred and vanished. They were encoded into your nervous system, transformed from fleeting patterns of neural activity into lasting changes in the connections between neurons. This transformation, the conversion of experience into memory, is one of the most remarkable processes in your brain. It allows the past to inform the present, enables learning from experience, and creates the continuity of self that persists across time. Sensory memory begins at the moment of perception. When you see, hear, touch, taste, or smell something, the neural activity in your sensory cortices does not simply process the stimulus and then return to baseline. Instead, that activity triggers a cascade of molecular events that can strengthen the connections between the neurons involved. If the experience is repeated, if it is emotionally significant, or if you pay attention to it, these molecular changes become more pronounced, making the neural pattern more likely to be reactivated in the future. The initial stage of sensory memory is extremely brief, lasting only a fraction of a second. This is called sensory or iconic memory for vision and echoic memory for hearing. During this fleeting period, a nearly complete representation of the sensory input is maintained in the sensory cortices themselves. You can experience this form of memory when someone speaks to you while your attention is elsewhere. You may not consciously hear the words initially, but if you redirect your attention within a second or two, you can often replay what was said. accessing the trace that briefly persisted in your auditory cortex. Most sensory information fades from this initial memory without further processing. Your sensory systems are constantly bombarded with information, far more than your brain could possibly encode into lasting memory. Selection occurs through attention. When you pay attention to something, the neural activity representing that stimulus is amplified and sustained, making it more likely to transition from sensory memory to short-term or working memory. Working memory is the system that holds information in an active state for seconds to minutes, allowing you to manipulate and use it for ongoing tasks. When you remember a phone number long enough to dial it, or when you hold a visual image in mind while drawing, you are using working memory. This form of memory relies on sustained neural activity in cortical networks, particularly involving the prefrontal cortex. The prefrontal cortex maintains representations of sensory information even after the stimulus has disappeared, keeping the information accessible for immediate use. For sensory experiences to become lasting memories, the kind you can recall days, years, or decades later, they must undergo a process called consolidation. Consolidation is the transformation of temporary, fragile neural patterns into stable, enduring changes in the brain's structure and function. This process involves the hippocampus, a seahorse-shaped structure deep within the temporal lobe that is essential for forming new long-term memories. The hippocampus receives input from all sensory modalities, though this input arrives indirectly through association cortices, rather than directly from primary sensory areas. When you experience something memorable, the pattern of activity across your sensory and association cortices is relayed to the hippocampus. The hippocampus binds these distributed patterns together, creating an index that links the various sensory components of the experience. When you later recall that experience, the hippocampus reactivates the pattern across cortical areas, bringing back the sights, sounds, smells, and other sensory qualities that defined the original event. The consolidation process unfolds over time, with the most active phase occurring during sleep. When you sleep, the hippocampus replays the neural patterns from recent experiences, repeatedly activating the cortical areas that were involved in the original perception. This replay strengthens the connections between neurons in those cortical areas, gradually making the memory less dependent on the hippocampus and more firmly embedded in the cortex itself. Eventually, well-consolidated memories can be retrieved without hippocampal involvement, though the hippocampus typically remains involved in recalling the rich contextual details of experiences. The strength of sensory memories depends on several factors. Emotional arousal powerfully enhances memory consolidation. The amygdala, a structure involved in processing emotion, modulates hippocampal activity during memory formation. When you experience something emotionally significant, whether joyful, frightening, or moving, the amygdala signals the hippocampus to prioritize consolidation of that experience. This is why emotionally charged events tend to be remembered more vividly and for longer periods than neutral events. Repetition also strengthens sensory memories. Each time you re-experience something similar, the neural pathways involved become more efficient. The connections between neurons that fire together are strengthened through a process summarized by the phrase, "neurons that fire together, wire together." At a molecular level, repeated activation causes changes in synapses, the junctions between neurons, making them more effective at transmitting signals. New proteins are synthesized, new receptors are inserted into cell membranes, and sometimes entirely new synaptic connections are formed. Sensory memories are often highly specific, preserving not just the general category of what was experienced, but precise details. You can distinguish between thousands of different faces, recognize a friend's voice from a single word, identify specific foods by smell or taste. This specificity arises from the fine-grained patterns of activity in sensory cortices that are preserved during memory formation. When you encode a memory of a face, the specific pattern of activity across face-selective neurons in your temporal cortex is stabilized through synaptic changes. Later, recall reactivates a similar pattern, bringing back the visual details of that particular face. Recognition is the process of perceiving something as familiar, of matching current sensory input to stored memories. When you recognize a friend's face or identify a familiar song, your brain is comparing the current pattern of sensory activity to patterns stored from previous experiences. Recognition occurs when there is sufficient overlap between the current pattern and a stored pattern. This matching process happens rapidly and often unconsciously, giving rise to the immediate sense of familiarity that accompanies recognition. The stored patterns that enable recognition are not perfect recordings of original experiences. Memory is reconstructive rather than reproductive. Each time you recall a sensory experience, your brain rebuilds an approximation based on the stored information, filling in gaps and sometimes introducing distortions. This is why memories can change over time, becoming less accurate or incorporating details from other experiences. Two people who witness the same event may remember it differently, not because one is lying, but because memory is an active construction, shaped by attention, expectation, and subsequent experiences. Sensory memories are organized by their relationships to other memories. When you smell coffee, you may automatically think of morning, recall a specific cafe, or remember a person you often had coffee with. These associations reflect the way memories are stored in interconnected networks. Experiences that occur together or share features become linked, so that activating one memory makes related memories more accessible. This associative organization allows for efficient retrieval, but also means that memories are not stored in isolation. Each is embedded in a web of related experiences. The interaction between memory and perception is bidirectional. Memory influences what you perceive, and perception updates your memories. When you encounter something familiar, your stored memories create expectations about what you will experience. These expectations can speed recognition and fill in ambiguous details, but they can also bias perception, making you see what you expect rather than what is actually present. This top-down influence of memory on perception demonstrates that what you see, hear, and feel is not a pure reflection of sensory input, but a construction shaped by prior experience. Certain sensory modalities have particularly strong connections to memory. Smell, in particular, is remarkably effective at triggering vivid autobiographical memories. This phenomenon, sometimes called the Proust effect, after the author Marcel Proust, who famously described how the taste of a Madeleine cookie evoked detailed childhood memories, reflects the direct anatomical connections between the olfactory system and memory structures. Unlike other senses that relay through the thalamus, olfactory information projects directly to the piriform cortex, and from there to the hippocampus and amygdala. This direct pathway may explain why smells can trigger such immediate and emotionally laden memories. Music also has a powerful ability to evoke memories. Hearing a song from a particular period of your life can instantly transport you back, bringing with it a flood of associated sensory memories, emotions, and contextual details. This effectiveness may reflect music's engagement of multiple brain systems simultaneously: auditory processing in the temporal lobe, emotional processing in the limbic system, motor areas that respond to rhythm, and memory systems that bind these components together. The capacity for sensory learning continues throughout life, though it is particularly pronounced during development. Infants and children are constantly forming new sensory memories as they learn to recognize objects, faces, voices, and patterns in their environment. The neural circuits for sensory processing are refined through experience, with connections that are frequently used being strengthened, and those that are rarely used being pruned away. This experience-dependent plasticity allows the brain to optimize its sensory processing for the specific environment in which it develops. Even in adulthood, sensory experience continues to shape neural structure. Learning new skills that involve sensory discrimination, such as wine tasting, musical training, or tactile reading, produces measurable changes in the relevant sensory cortices. The cortical representations of frequently used sensory features become enlarged and more precisely tuned. Expert wine tasters show enhanced activity in olfactory and gustatory areas when evaluating wines. Musicians show enlarged representations of the fingers in their somatosensory cortex. These changes demonstrate that sensory cortices remain plastic throughout life, capable of reorganization in response to experience. Sensory memories can persist with remarkable clarity for decades. You may be able to recall the sound of a grandparent's voice, the appearance of a childhood home, or the taste of a favorite food from years past. These enduring memories reflect stable changes in cortical circuits, patterns of synaptic connectivity that have been maintained across time. Yet even long-term memories are not entirely permanent. Without occasional reactivation, memories can fade, losing detail and vividness. Each time you recall a memory, you partially reactivate and potentially modify it, either strengthening it through reconsolidation or subtly altering it as new information is incorporated. The loss of sensory memory is one of the most distressing aspects of aging and neurological disease. Alzheimer's disease, in particular, affects the hippocampus early in its course, impairing the ability to form new memories while often leaving older, well-consolidated memories initially intact. As the disease progresses, even remote sensory memories can be lost, erasing a lifetime of accumulated experiences. The preservation and retrieval of sensory memories, the sights, sounds, and smells that define a life, represents one of the most important functions of your brain. Right now, your brain contains countless sensory memories. The neural patterns encoding these memories are distributed across your cortex, embedded in the precise patterns of synaptic connections between billions of neurons. Some of these memories are actively accessible, readily retrieved when you think about them. Others lie dormant, not thought of in years, but still present, waiting for the right cue to bring them back to consciousness. The sound of a particular song, the scent of a specific flower, the sight of a familiar place. Any of these could trigger the reactivation of memories you had forgotten you possessed. The relationship between sensory memory and imagination reveals that perception and memory use overlapping neural machinery. When you imagine seeing something, you activate many of the same visual cortical areas that would be active if you were actually seeing it. When you imagine hearing a melody, your auditory cortex becomes active. The patterns are not identical to those produced by actual perception. Imagination tends to produce weaker and less detailed activity, but the similarity is striking. This overlap means that remembering a sensory experience is, in a sense, re-experiencing a weaker version of it. Dreams draw heavily on sensory memories. During sleep, especially during rapid eye movement sleep, your brain spontaneously activates patterns in sensory cortices, creating vivid perceptual experiences without any external input. The sights, sounds, and sensations in dreams are constructed from fragments of stored sensory memories, recombined in novel and often bizarre ways. The content of dreams reflects recent experiences, long-term memories, and emotional concerns, all filtered through the unique logic of the sleeping brain. Memory also plays a crucial role in perceptual learning, the improvement in sensory discrimination that comes with practice. When you first encounter a new perceptual task, such as distinguishing between similar sounds or identifying subtle visual differences, performance is poor. But with repeated exposure, your ability improves dramatically. This improvement reflects changes in sensory cortices, where neurons become more selective for the features that are relevant to the task. The stored memories of previous encounters. With these stimuli guide neural tuning, optimizing sensory processing for the distinctions that matter. Pattern completion is another important function of sensory memory. When you encounter a partial or degraded sensory stimulus, your brain automatically fills in missing information based on stored memories of similar experiences. You can recognize a face from a brief glimpse, identify a word from partial letters, or recognize a song from a few notes. This completion process relies on the associative nature of memory. When part of a stored pattern is activated, the entire pattern tends to be retrieved. Pattern completion allows you to perceive coherent, meaningful experiences even when sensory input is incomplete or ambiguous. The emotional coloring of sensory memories adds another dimension to their retrieval. Memories are not stored as neutral records but are tagged with emotional significance. When you recall a sensory experience, you often re-experience some of the emotion that accompanied the original event. A smell associated with a happy time evokes not just the memory but also a faint echo of the happiness itself. Conversely, sensory cues associated with negative experiences can trigger anxiety or aversion, even when the original context is no longer relevant. This emotional dimension of memory influences your behavior, guiding you toward positive experiences and away from negative ones based on past outcomes. Memory reconsolidation is a recently discovered phenomenon that has changed how neuroscientists think about memory stability. When you retrieve a memory, it becomes temporarily unstable, susceptible to modification, or even erasure. During this reconsolidation window, the memory must be restabilized through molecular processes, similar to those that occurred during initial consolidation. This instability means that memories are not fixed records, but are reconstructed and potentially altered each time they are recalled. Therapeutic interventions are being developed to target reconsolidation, potentially allowing modification of problematic memories, such as those underlying post-traumatic stress disorder. Sensory prediction is deeply intertwined with memory. Your brain uses stored memories to generate predictions about what you are likely to experience next. When you hear the first few notes of a familiar song, your brain predicts what will come next. When you reach for an object you have touched before, your brain predicts what it will feel like. These predictions are not conscious hunches, but automatic processes that shape neural activity in sensory cortices before the predicted input arrives. When predictions match reality, perception is facilitated and requires less processing. When predictions are violated, prediction errors are generated, which drive learning and update your stored memories. The accumulated sensory memories of your lifetime shape your identity and your understanding of the world. The faces you recognize, the places you know, the sensations you can name and distinguish. All of these capacities depend on the vast library of sensory patterns stored in your cortex. Without memory, each moment would be isolated, disconnected from past and future. With memory, experience accumulates. Patterns emerge, and the continuity of self persists across time. As you rest here now, sensory information continues to flow into your brain. Some of this information is being encoded into memory, strengthening existing patterns or creating new ones. Most will fade without trace, never consolidated into lasting storage. But the memories that do form will become part of the vast network of stored experiences that define who you are and how you perceive the world. All of this, the sensation, the processing, the memory, culminates in something that science still struggles to fully explain. All of these neural patterns, all of these electrical signals and synaptic changes somehow give rise to subjective experience, to the feeling of what it is like to see, to hear, to feel. From the mechanics of sensation and memory, we turn finally to the deepest mystery. How does the physical activity of your brain create the conscious experience of being you? You experience the color red as having a particular quality, a redness that seems irreducible and unanalyzable. You experience pain as feeling a certain way, unpleasant and urgent. These qualitative aspects of experience, what philosophers call qualia, seem fundamentally different from the neural processes that produce them. An outside observer studying your brain could potentially measure every aspect of the neural activity occurring when you see red or feel pain. They could trace the signals from receptors through pathways to cortex, document every spike, every synapse, every pattern of activity. But would they thereby know what it feels like to be you, experiencing red or pain? This gap between objective description and subjective experience is what makes consciousness the hard problem. Despite this mystery, neuroscientists have made substantial progress in identifying the neural correlates of consciousness, the specific patterns of brain activity that correspond to conscious experiences. The search for these correlates begins with a fundamental distinction. Not all neural processing is conscious. Much of what your brain does happens without your awareness. Your visual system performs extensive computations to extract edges, detect motion, and analyze colors, yet most of this processing never reaches consciousness. Similarly, motor control involves complex calculations about force, trajectory, and timing, but you experience only the intention to move and the result, not the intermediate computations. What distinguishes conscious processing from unconscious processing? Research suggests several key factors. First, conscious perception requires sustained neural activity. Brief, transient activation of sensory cortices is not sufficient for awareness. The neural response must persist for at least 100 to 200 milliseconds, and it must be amplified through feedback connections. Second, conscious perception involves widespread activity across distributed brain networks. While unconscious processing can be localized to specific sensory areas, conscious experience activates large-scale networks spanning multiple cortical regions. The thalamocortical system is central to consciousness. The thalamus not only relays sensory information to the cortex but also regulates which signals reach awareness. During anesthesia, thalamocortical activity is suppressed and consciousness disappears, even though sensory signals continue to arrive at peripheral receptors and lower processing stages. This suggests that the thalamus acts as a gatekeeper, determining which sensory information will be broadcast to widespread cortical areas and thus become conscious. The prefrontal cortex also appears essential for conscious awareness. This region, which is involved in attention, working memory, and executive control, shows consistent activation during conscious perception. When you are aware of a stimulus, your prefrontal cortex becomes active, integrating sensory information with context, goals, and expectations. Damage to the prefrontal cortex can impair certain aspects of consciousness, particularly the ability to reflect on and report one's experiences. Attention is intimately related to consciousness, though the two are not identical. You can attend to something unconsciously, and conversely, you can sometimes be conscious of things you are not actively attending to. However, attention and consciousness typically go together. When you pay attention to a stimulus, the neural activity representing that stimulus is enhanced in sensory cortices, making it more likely to be amplified into widespread cortical networks, and thus to reach consciousness. Attention acts like a spotlight, selecting which aspects of sensory input will be processed deeply enough to become conscious experiences. Several theories attempt to explain how neural activity gives rise to consciousness. Global workspace theory proposes that consciousness arises when information is broadcast to a widespread network of brain regions, making it available for multiple cognitive processes simultaneously. According to this theory, unconscious processing is modular and localized, while conscious processing involves global integration. When sensory information enters the global workspace, implemented by widespread cortical networks and coordinated by prefrontal cortex, it becomes conscious and can be used for reasoning, decision-making, and verbal report. Integrated information theory takes a different approach, proposing that consciousness corresponds to integrated information, the degree to which a system is both differentiated and unified. A conscious system must be able to exist in many different states, providing differentiation. But these states must be integrated such that the system functions as a unified whole. The theory attempts to quantify consciousness using a measure called phi, which represents the amount of integrated information in a system. According to this theory, any system with sufficiently high integrated information would be conscious, regardless of whether it is biological. Recurrent processing theory emphasizes the importance of feedback connections between brain areas. According to this view, consciousness arises not from the initial feed-forward sweep of sensory processing, but from recurrent interactions between higher and lower cortical areas. Visual information reaches V1 within 40 to 60 milliseconds, but consciousness of that information requires feedback from higher visual areas and prefrontal cortex, which takes an additional 100 milliseconds or more. These recurrent loops amplify and sustain neural activity, transforming brief sensory responses into stable conscious percepts. The binding problem is another puzzle in understanding consciousness. When you look at a red circle, different features, color, shape, location, are processed in different brain areas. How does your brain bind these distributed representations into a unified conscious experience of a single object? One proposed solution is neural synchrony, when neurons representing different features of the same object fire in synchrony, oscillating together at particular frequencies like gamma oscillations around 40 Hz. They may become bound into a unified representation. This synchronized activity could tag neurons as belonging to the same perceptual object, solving the binding problem. Consciousness is not all or nothing but comes in degrees. You can be more or less alert, more or less aware of your surroundings. During sleep, consciousness dims but does not entirely disappear. Dreams are conscious experiences occurring in an altered state. Under anesthesia, consciousness can be completely abolished, with no experience and no memory of the period of unconsciousness. These variations in consciousness correspond to changes in the patterns and extent of neural activity across the brain. The neural correlates of specific conscious contents, what you are experiencing at any given moment, can be identified with increasing precision. When you consciously see a face, specific face-selective regions in your temporal cortex become active. When you consciously experience pain, the anterior cingulate cortex and insula activate. When you are aware of your own thoughts and mental states, what is called metacognition, the prefrontal cortex shows characteristic patterns of activity. By comparing brain activity during conscious versus unconscious processing of the same stimulus, researchers can isolate the neural signatures of awareness. Binocular rivalry provides a powerful tool for studying consciousness. When different images are presented to each eye, you do not see a blend, but instead experience alternations. First one image is conscious, then the other. The sensory input remains constant, but your conscious perception switches back and forth. By measuring brain activity during these switches, researchers can identify which neural changes correlate with changes in conscious experience, independent of changes in sensory input. Studies show that when perception switches, activity shifts between different populations of neurons in visual cortex, and this shift propagates to higher cortical areas. Blindsight demonstrates that sensory processing can occur without consciousness. People with damage to primary visual cortex lose conscious vision in the corresponding part of their visual field. Yet when asked to guess about objects in that blind region, they perform better than chance, revealing that visual information is still being processed unconsciously. This dissociation shows that not all sensory processing leads to awareness, and that V1 may be particularly important for conscious vision. The unity of consciousness is another remarkable feature that demands explanation. Despite the vast amount of sensory processing occurring in parallel across different brain regions, you experience a single, unified stream of consciousness. You do not experience vision separately from hearing or touch separately from proprioception. Instead, all sensory information is integrated into a coherent experience of a unified world. This unity must arise from the coordination and integration of activity across widely distributed neural networks.